Good morning, everybody. It is Tuesday, June 23rd. Hopefully you survived the lab and lecture exam yesterday, and we are moving on to the second part of the class, our skeletal system. It, as you can see, is a short turnaround. It's a short section, but it has a lot of material in it, so we are going to be very, very busy. Today we will start our discussion talking about skeletal tissue, basically both the microscopic and gross anatomy of skeletal tissue, and then setting the foundation for what we'll be doing for the rest of the time for this section. That is going to be talking about the physiology of bone in the lecture. I know, again, we keep talking about when you see bones, you're usually seeing them as a skeleton and it is dead, and we think of it as dead, dried up material. In fact, that's what skeletos means. Skeleton comes from the word skeletos, which means dried up. But as we will learn, bone is an incredibly dynamic tissue with a lot of physiology to it. So after today, pretty much what we'll be doing in lecture is talking about the physiology of bone and skeletal tissue, how we make bones, how we grow bones, how we heal bones, how we maintain bones, all of those types of processes. And then in lab, we will be working together to learn the bones and bone features you are going to be responsible for. I'm not going to make you do it on your own. You'll be forming groups of 2 or 3 people. We'll be forming those groups today. I will be doing breakout groups during the after the end of lecture today, and I will also be setting up a discussion board for your group so that you guys can communicate asynchronously as well. Although when we first break up into groups, I would encourage you to at least share email addresses, if not other contact information, so you guys can work together on this. What you're going to be doing is presenting bones and bone features, and I will talk about that process and what you're going to be doing on that. It's very simple, very straightforward, very low pressure. So it isn't anything to worry about that way. But we'll talk about that more when we get to it. However, the other thing that I think will be useful, since I'm breaking you up into groups anyway, as you see on the assignments, there is a 30-point skeletal review that is going to be due on Tuesday, June 30th. This one is going to be your first major assignment that is going to be graded for correctness. So this is a homework assignment, but this time it is going to be graded for correctness, which means that wrong answers lose you points. Towards that end, I think it would be useful. I found it useful in the past where students often work together on this when they're in the classroom, when they're in the lab. So I'm going to encourage you as groups to work on these together as groups as well. Help each other out, discuss these things to try to come up with the correct answers. However, I still want each student to turn in their own individual assignment, right? So when you're doing this, I want you guys to discuss it together, come up with the right answers. But when you're writing out the answers, I want you writing them out in your own words. I don't just want one copy of it with everybody's name on it, or I don't want everybody's answers to be word for word identical. Now, that being said, I will point out that some of the answers to questions are things like cervical vertebrae. Right? If you're just writing out cervical vertebrae, then fine. Yeah, there's no other way to write that. You'll just write cervical vertebrae. But however, if you have to, for instance, describe some process or describe some physical characteristic, I don't want your 3 sentences of description of that to be word for word the same as everybody else's. You can come up with your right answer together, but decide, how you are going to write that in your own words. If you're asked to describe what today's weather is going to be, right, I would say that it is very, very hot, whereas you could say that it is uncomfortably warm, right? We can all say the same thing, but do it in our own words. So that's what I'm looking for in that. But like I said, if the answer is cervical vertebrae, then the answer is cervical vertebrae. You don't have to reword something like that. All right. You will notice, hopefully you have noticed, because you have gotten onto Canvas and looked at the new module, although we will do that together again a little bit later as well, that the skeletal review is not available yet. Because the point of this skeletal review is to review the skeleton. What we are going to be doing as a group is looking at each individual bone and identifying all of the key little characteristics, the bumps and nooks and crannies on it, and being able to identify all of the bits and pieces. But what we have to remember is that these bones fit together into a skeleton to form a fully functional organ system that is the skeleton. And that is what this skeletal review is about. Part of the review is understanding how the bones relate to each other, to understand that the head of one fits in the cavity of another. If you don't know the name of that bone feature and you don't know the name of this bone feature, then knowing that these two bone features relate to each other isn't a meaningful piece of information. So you'll notice that the skeletal review is not available for you yet, because what I want you doing first is focusing on the bones and bone features. And if you do that first, then you'll be more successful at putting it together. I do this on purpose because what I find is that when people see there is a 30-point assignment that they're going to be doing, they rush to try to do it and get that done because they think it is important, and it is important. But if you don't know the bones and bone features, you're not going to understand how the bones relate to each other. So it's not going to be, it's going to be a frustrating activity. It is going to be a challenging activity, and it is going to take you much more time than it would take you otherwise if you know the bones and bone features first. If you know the bones and bone features of the bones, how they fit together is going to be make much more sense, and it'll be a much easier assignment. So I've got that locked for a couple of days to force you to think about the bones and bone features first. Then you can start focusing on the skeletal review. And again, I encourage you to use your discussion board, use your contacts information to be able to go through that. All right. There are 3 other assignments to do during this section, and those are the Unit 7, Unit 8, and Unit 9 reviews. In grading this weekend, I noticed that some of you are still missing all of the assignments. I try to put the page number there to help you, but the simple rule to remember is when you look at these reviews have 2 parts, a check your recall and a check your understanding. And so both parts of those reviews are due. And again, make sure you are putting time and effort. Remember, I'm not grading you for correct answers on this, but I am for time and effort. So if the question asks you, whether something is going to be A or B, and explain your answer, and all you say is A, that doesn't show me time and effort. And again, starting this week, you're going to be losing points for turning these things in late, and it is important to make sure you are filling these things out completely and correctly and doing them properly. All right? All this leading up to the 4th of July weekend, right? July 2nd is our lab and lecture exams. So again, you get to take that, and as soon as you're done with that, go watch Hamilton. All right, so again, the nice thing about that is that you've taken the lab and lecture exam. There are going to be no surprises. You know exactly what to expect. The exact same format for the lab exam is going to be the exact same format for all your lab exams. The exact same format for your lecture exam is going to be the exact same format for all your lecture exams. So there should be no surprises. You should know what to expect, especially now. I've only started grading, but I can already see that this first test, hopefully, has been a wake-up call for some of you. Some of you did not prepare nearly as well as you should have. Well, again, I've looked at the lecture exam. I haven't looked at any of the lab exams, and I've just started. But I can see that some of you really struggled with this information. And again, as I've mentioned, there are 2 things you have to do to be successful in a class like this. Not only do you have to cram all the information into your brain, but you also have to present that information on the exam. You have to be descriptive. You have to use words. You have to describe things. It's in the question. Identify and describe things. I've written so many, describe, explain, expand, right? Be more descriptive. Make sure you are doing those things, because that is how you're going to maximize your points on these. And we'll talk about the tests more when I return them. You will, once they're graded, you will get an opportunity to see them so that you can learn from them. And we'll talk about that process when we get closer to it. On average, it usually takes me about a week to grade all of the exams with both labs and lecture exams. I will work as hard as I can and try to get it done as quickly as possible. The latest you would get it back is Monday. But honestly, I'm hopeful to try to get it back by Friday at least, so that we can talk about it on Friday before we go into the weekend. All righty. Any questions on any of that? All righty. Then let's go ahead and begin. So we have moved on to our second organ system, and that second organ system is the skeletal system. As I mentioned, what we need to do first is talk about our bone tissue. As I already mentioned, this word skeleton comes from the Greek word skeletos, which means to be dried up. But as we already talked about, the bone is a very dynamic connective tissue, making the bones a very dynamic organ. The skeletal system is an organ system. So it, of course, includes organs. And what did we say the organs of the skeletal system was? Oh, sorry, I don't have my chat.
Bone.
There you go. Excellent. There, the bones, exactly. And how many bones did we say again?
206.
Excellent. Although we do, as we'll see now, we do want to be a little careful with this. There are 206 named bones. As we will learn as we move forward in today's lecture, your skeletal system actually has far, far more than 206 bones in them. However, apparently after naming 206 of them, they got tired and so didn't bother naming any of the rest. So we have 206 named bones, and as we've talked about, you are absolutely responsible for all of those. Bones are indeed an organ. What was our requirement to be an organ again? How do we make organs?
Tissue.
True, absolutely. And how many different types of tissues did we say you have to make an organ? 2 or more, excellent. We have to have 2 or more different tissues. And we absolutely have that in bones. Obviously, as the name indicates, our bone connective tissue is the most common connective tissue that forms the organ that we call the bone. But as we will learn, there is also, as we've already talked about, we know the ends of the bone have hyaline cartilage. As we will see, there is a dense, irregular connective tissue. That lines the outer surface of much of the bone. And that doesn't even include things like the nerves and the nervous tissue that are running inside of them, the blood vessels that are running inside of them. So obviously they're clearly made up of lots of components and absolutely positively are indeed organs. We already did the numbers, so we'll talk about that later. And again, as we've talked about, the study of the bones is the field of osteology. All right, with that in our pocket then, let's talk about the organ system. And as we said before, often when we discuss an organ system, we want to identify the functions of that organ system. And conveniently enough, as it sees here, there are 6 specific functions that our organ system does for us. So give me an example of one of the functions of our skeletal system.
Support.
Excellent, support. Right? Absolutely. Its job is to give basically the vertical axis to the body, right? Gives us that support, allows us to stand up. What else?
Movement.
Oops. Screw that up. Yep. Absolutely. Movement. Perfect. They are the levers that our muscles pull on to move us through space. What else?
Next, blood.
Excellent. Perfect. One of the things that it does is it is responsible for the formation of all the formed elements. Now remember, as we talked about, the formed elements are the red blood cells, the white blood cells, and the platelets. What component of the bone is responsible for the formation of all of the cells of the blood?
The marrow, the bone, or the inside of the bone.
Excellent. You're absolutely right with the bone marrow. Although, could we be a teeny bit more specific? What type of marrow?
Red bone marrow.
There we go. That's what I was looking for. Red bone marrow. Excellent. It is the red. Oops. I spelled red right. It is the red bone marrow that is responsible for forming all of the cells of the blood, all of the formed elements of the blood. Excellent. What's another function?
Protection.
Protection, absolutely. Oops, again, wow. Protection, absolutely. All right, support, movement, protection. Those are the big three that everybody always thinks of. I love that the blood cell formation snuck in before that, because usually I always get the first three first, the big three first. That's excellent. 4 through, 2 more to go. What else does our bone do for us?
Does it store nutrients and materials?
Storage, absolutely. But again, just like before, we can probably be a little bit more specific. Storage of what?
Calcium.
There you go, absolutely. Calcium is a big one, but let's start a little less specific. Minerals in general, but you're absolutely right. The big one for that is calcium. Something like 99% of the calcium in your body is stored inside of your bones. And as we talked about, calcium makes cells do wonky things. As we'll see, we have to keep a very tight, precise control of how much calcium is in our blood. So it is so important for maintaining appropriate levels of calcium in the blood that, again, like you guys, I'm sure, have no problem with this. You all had a nice and big breakfast, right? You had that nice big cheeseburger that had cheese on it, a nice big glass of milk with it, and got all the calcium that you needed. But some people have a donut and a Diet Coke for breakfast. And are they necessarily getting the calcium that they need from their diet? No. And if we're not getting the calcium that we need in our diet, then we basically have two choices. We can have big, strong bones with lots of calcium in them, or we can start breaking down that bone to make sure we have enough calcium in the blood. And if it's weighing those two options against each other in our body, which one's going to win all the time? Calcium in the blood or calcium in the bones? What do you think is more important to your body?
Bones.
It's actually calcium in your blood. It's calcium in your blood that's going to be more important.
Breaking it. Gotcha.
Right, exactly. So if need be, our body will actually break the calcium out of our bones, weaken our bones, to make sure we have an appropriate amount of calcium. But it's not just calcium, it's phosphates, magnesium. There's all sorts of other minerals that are in there. And where in the bone do we store these calcium, these phosphates, these minerals?
In the articular cartilage? Or is it the... Because it's above the trying to go back to my 100 notes.
No, you're no problem.
It's above the epiphyseal line.
Yeah.
Where it stores, and that's not the shaft. Somebody else take it. I can't.
You're actually right along. You have the right idea. It is predominantly within the shaft of the long bones, at least if we're talking about long bones. But if you think about it, I think you're trying to be too specific in this case. If you think about where the calcium, where the phosphate, where the minerals are stored is within the matrix of the bone connective tissue. Right? Remember, our bones are primarily made up of bone connective tissue, and like all connective tissues, they're made up of cells and matrix. So it is the matrix of the bone connective tissue where we have the calcium crystal salts and things along those lines that are going to be stored. So that's where we're storing the minerals. Notice we had to be specific about storing minerals because there's something else that is stored within our bones as well. What is our sixth function of the bones is the storage of what?
Triglyceride.
Excellent. Triglycerides, right? Fats. Primarily triglycerides. Or I guess fats is bad. Let's at least say lipids. But you're absolutely correct. The primary lipid that is stored are those triglycerides. And where do we store the triglycerides inside of our bones?
Yellow marrow?
Excellent, perfect. That's kind of why we had to say red bone marrow before, because as a mature adult, right, if you've got a little kid and you sucked all the marrow out of their body, all of the bone marrow would be red. But if I sucked all of your bone marrow out of your body, would it all be red? No, in fact, most of it would be yellow bone marrow. Actually, the places that have red bone marrow are more restrictive in an adult. So a lot of the marrow in our bones become yellow bone marrow. Now, why? Why do we store lipids in our bone marrow, in our bones?
for a source of energy?
Absolutely. As we know, lipids are a source of energy, so we can store it there. Excellent. Our bone marrow has got a lot of stuff to do, so having a lot of energy could be useful. Why else might we store bone marrow in the center of our bones? I mean, yellow bone marrow in the center of our bones. Well, we're further away from January, so you don't see these things as much anymore. But one of the things you see typically at the first of the year, because that's when everybody's trying to, make their New Year's resolutions. That's when everybody signs up for the gyms and all those things. And they love to have those commercials where someone holds up a pound of muscle and a pound of fat. And when they hold up a pound of muscle and a pound of fat, are they the same size? Which one's bigger?
Fat.
Fat. Fat is a very light, loose material. So one of the advantages, especially in our large, long bones, of filling the cavities of them with yellow bone marrow is it decreases the overall weight of the skeletal system as well to make it more easy to move around. So it also helps to maintain homeostasis by making that yellow bone marrow in there, lightening up the bone and making it easier to move around in space. Excellent, excellent, excellent. So we've hit all 6. Let's clear my writings of them and look at the pretty pictures of them. So like we said, the big 3, support, protection, and assist in the movement, provide the leverage for the skeletal system, for the muscular system. Right. Hematopoiesis, which again, as we talked about, is the fancy name for the formation of all formed elements in the blood. And again, anytime you see an alphabet soup word like that, you can be fairly guaranteed you're going to have to spell that at some point. So you definitely need to know that fancy term. And again, as we talked about, that is going to occur in our red bone marrow. We have our mineral homeostasis, storage of calcium, storage of phosphoruses, storage of magnesium, other things along those lines. And as we said, that is going to occur within the matrix of the bone connective tissue. That's what I've heard. And lastly, our triglyceride storage, right? And again, this is found in our yellow bone marrow. Right. That fat storage of energy, it provides some shock absorption to the bone, helping to provide some internal support, but it also helps to lighten the load of the bone as well. So it is a little bit more functional, a little bit more feasible to be moving it around, maintaining homeostasis. All right, questions on any of that? All right, perfect. Then let's talk first about our bone connective tissue, or again, remember when we talked about bone connective tissue. Since bone connective tissue and bones organs can be confused. Remember, we also said that we can use the term osseous tissue, and again, both are acceptable. Basically, with like all connective tissues, we have cells, and those cells are surrounded by a large amount of matrix, right? And in our bone connective tissue, there are primarily 4 main types of cells. Now, before we talk about the specific cell types, I do want to do a little bit of discussion first on the basic anatomy of what we're seeing here with the bone. Notice what they've done here is kind of a cross section through a piece of bone. So I want to take a little bit of a closer look at that first before we start talking about the 4 cell types we find inside of our bone connective tissue. So let's switch back to the whiteboard. Clear the notes from earlier and do a little drawing. So we'll take that same image. Here, hold on, let's undo that. I'll make it black. There is the outer surface of our bone. And notice the inner surface of our bone had more of a rough design to it. And then, of course, this here is the cavity at the center, the medullary cavity, where, as we mentioned, you're going to find the bone marrow. And I'll just abbreviate that BM for right now. All right. Now, we know that most bone of the body is compact bone. And if you remember correctly, we said compact bone had these organizing structures. What did we say the organizing of compact bone was again? It was a circular structure with a hole at the center.
Oh, the central thing. Osteon?
Osteon, excellent. That organizing structure of the compact bone is indeed an osteon. Right? And that has a central canal at the center, and it has the circles around it and the spaces for the bone and some of the anatomy that we already talked about, and we'll talk about those things in more detail as well. But there's a couple other things. Remember, we said bone has blood vessels and nerves that pass through this central canal. But remember, I also told you that this tissue has and let's use a different color. I want to do that, and I want to do that. Bone has a dense, irregular connective tissue that wraps around the outer surface of the bone. Now, again, my drawing skills aren't that great, and I purposely left space there because there's some things we want to draw in here. But typically, it is right on the surface of the bone. This tissue happens to be a dense, irregular connective tissue. Right, that is its tissue type. However, this isn't the only place we find a dense irregular connective tissue in the body. Right? In fact, if I remember correctly from the last exam, there was a dense irregular connective tissue that wrapped around cartilage. And when it wrapped around cartilage, we gave it a name based on its location. And what was the name we gave that dense irregular connective tissue that wrapped around the outside of our cartilage?
Fibrocartilage?
What did we call the dense irregular connective tissue that wrapped around the outside of our hyaline cartilage? You guys are not just a lot of faith for when I grade the lab exams. Well, we called it a perichondrium. Oh, there we go. Sandy got it. Perichondrium. Excellent. Perfect. Well, this is a dense irregular connective tissue that wraps around bone. So if a perichondrium wraps around bone, guess what we call a dense irregular connective tissue based on its location that wraps around bone?
Perichondria?
Well, peri means around, like perimeter. Chondrium refers to cartilage. So in this case, since it's around bone, it would be a periosteum. Osteum. Excellent. So we have this periosteum, this dense irregular connective tissue that wraps around the outer surface of the bone. And as I mentioned, I purposely left some spaces there. And the reason I purposely left those spaces is because, as we're going to see, there are going to be some immature cells that will be found housed inside of our periosteum. So this periosteum acts as an attachment point for the tendons and ligaments. It is going to house the immature cells and stabilizes blood vessels. And it also helps to protect the bone. Excellent. Now, interestingly, it also turns out that there is a dense, irregular connective tissue that lines the inner surface of our bone as well. And again, I'm purposely leaving a space, but normally it is in direct contact. And again, some of that is because of my poor drawing skills, and some of it is because we need that space to put something in it. this is also a dense, irregular connective tissue. However, this one is on the inside of the bone. And since it's on the inside of the bone, we can't call it a periosteum because peri, like perimeter, means on the outside. So what is a term that means on the inside? It's a term, what's a prefix that means inside of, within? True, visceral means within, absolutely. But remember when we talked about glands, we had glands that secreted outside the body and glands that secreted inside the body. What are the glands that secrete inside within the body called again? Endocrine, excellent. So endosteum, perfect. All right, fun with vocabulary. Endosteum is what we call this dense irregular connective tissue on the inside of the bone. Now, if we think of the three functions of the periosteum and attachment points for tendons and ligaments can't attach on the inside of the bone. Surrounding and protecting really isn't something it can do on the inside of the bone. But what this will do is house immature cells. So there will be immature cells that we will find here inside the endosteum as well. So our endosteum and periosteum both house some of the immature cells that we are going to see on our bone in its microscopic anatomy. All right, so if we go back to the lecture, well, questions on this before I leave the page? All right, let's go back and look at the pretty picture from your textbook because it does a nicer job of showing these things. So here we have that picture from your textbook. Again, notice it doesn't show us our periosteum and our endosteum, but it does show us these basic organizations and locations. And with this, what we can do now is talk about the four main types of cells we have in our bone connective tissue. The first are what are known as osteogenic cells. Osteogenic cells, oh, I lost my annotation, there we go. Osteogenic cells are unipotent stem cells. Remind me again what it means to be a unipotent stem cell? Excellent. These are stem cells, so they're undifferentiated cells that divide rapidly, and these osteogenic cells can become one thing and one thing only. Of course, they come from our mesenchymal stem cells, which remember are pluripotent stem cells. So our mesenchymal stem cells divide to become osteogenic cells. And these osteogenic cells are the only bone cell that is capable of dividing. So once our bone cells start to mature, they are incapable of dividing. These osteogenic cells are the only bone cells that are capable of dividing. So that's mesenchymal cells. Remember, mesenchymal cells form all of the connective tissue cells. They divide to become osteogenic cells. And those osteogenic cells are the only bone cells that are capable of dividing. These cells are housed both inside the periosteum and the endosteum. So those little red dots we put, many of them are these osteogenic cells. And like I said, when they divide, they become one thing and one thing only, and the one thing they become is an osteoblast. Remember, osteoblasts, as we talked about, are the immature cells. And remember, immature cells, as we talked about, their job is to build the matrix. So B for blast, B for build. Our osteoblasts are the ones that are going to build matrix. All right. Questions on that?
Question, you said that once they mature, they cannot divide.
Yes.
Before then, they will.
Well, once they differentiate, really. So an osteoblast is not capable of division. So even though it's not a mature cell, it's not capable of division. So of the 4 bone cells we're going to talk about, the only one that is capable of dividing are these osteogenic cells. So they're unipotent stem cells. The osteoblasts cannot divide because, remember, as we talked about, division requires a lot of energy, and the osteoblasts are using all of their energy to make matrix. So that's why they're not able to divide.
Gotcha.
All right, great question. Any others? All right, excellent. Then let's talk about these osteoblasts. Notice they're kind of chunky little cells. They're osteoblasts, they're the immature cells, and their job is to, like we said, B for blast, B for build the matrix. This process of making the matrix is a process we call osteogenesis. Osteo, of course, still means bone. Genesis means to give birth to. Basically, what our osteoblasts do is they basically, they're surrounded by interstitial fluid. All right, so here, let me actually write some of this out. These cells are inside tissue, in the bone tissue, and they're surrounded by interstitial fluid. From that interstitial fluid, basically what they do is they grab the calcium and the other minerals that they need, and they basically form crystals, and they form those crystals around something. I know some of you have kids, and so this may be an activity you did or they actually did at home, or maybe you've done it yourself. Has anyone here ever made rock candy before? Anyone here made rock candy? Okay, anybody here know what rock candy is? Excellent. Okay, I got a couple yeses. Excellent. All right. So, how do you make rock candy? What is it that you do when you make rock candy? Does anybody remember how you do it? All right, I'll explain. Basically, you take some water and you pour a ton of sugar into it, right? Because the sugar is what's going to crystallize. And you heat it up. When you heat water up, as I'm sure you remember from chemistry, it allows more of the sugar to be dissolved into the liquid. And then you let it cool, at which point it's basically supersaturated. It's supersaturated with sugar. But as long as you leave it alone, nothing happens. But when you disturb it, like by putting a string in there, that string gives it a surface that those crystals can form and attach on. And so what happens is all these crystals start to form on the outer surface of the string. And that's exactly what happens here. Our osteoblasts take all these crystals and they're going to form all these minerals, and they're going to form a crystal out of these minerals, but they have to form the crystal on something. And so what happens is they form the crystal, and when they form the crystals, they actually form it on collagen fibers. So our bone has to have those collagen fibers in it so that those crystals can be formed on there and we can make our matrix. So that is what is going to happen. We initiate that calcification, we make those crystals. All right? Now, here's what happens. Let me now erase, actually, let me just move this up and there out of the way. And let's take an up close look at the side of our bone. So here is the edge of our bone. Let's just go ahead and put the edge of the bone there, right? And then we have our perichondrium, which I'm going to cheat and put way away from the bone. And then under here, we can put our osteogenic cell. So that's our original OG. right there. It, as we talked about, is going to divide, and when it divides, it forms a new cell. That new cell is our chunky osteoblast, which I'll write up here, OB. And as we mentioned, what it's going to start doing is it is going to start taking the calcium, taking the phosphate, taking the magnesium, taking the carbonates, taking all these things and making matrix. And then it makes a little more matrix, and it makes a little more matrix, and it's making matrix on this side, and it's making matrix on this side, and it keeps continuing to make matrix until this cell is completely surrounded by matrix. Once the cell is completely surrounded by matrix, there really isn't any place for it to make more matrix. And so our osteoblast is going to mature into an osteocyte. And what would we call this space that the osteocyte is sitting in? Lacuna, excellent. So it basically, once it surrounds itself with matrix, it now matures into an osteocyte, living inside of that lacuna, living inside of that cave that it made for itself. And we've just made a little bit more bone matrix. And then the same thing happens over here, and the same thing happens over here. And in this fashion, we are able to continue to add more and more layers of matrix on the outer surface of our bone. And our bone just got a little bit bigger. All still under the periosteum. All right? Questions on that?
So we said that when the osteocyte becomes the osteoblasts.
Nope, other way around.
Yep.
Osteoblast is the immature cell that builds matrix, and it surrounds itself with matrix. If you think about it, once it surrounds itself with matrix, then there's no more place to make more matrix. It can't continue to make matrix. And if it can't keep continuing to make new matrix, then it has to mature into the osteocyte. And remember, osteocytes' jobs are to maintain the matrix, right? So the mature cell's job is to maintain the matrix. Once it surrounds itself with matrix, it's going to maintain that matrix as a mature cell. But it really is just because of the location. Because it's surrounded by matrix, it can't keep making more. If you, for instance, break a bone, and when you break the bone, that osteocyte is released from its lacuna, it will actually revert to an osteoblast and will actually start making new matrix again. So it's really just because it's locked inside that matrix, it's locked inside of its lacuna, that it matures into an osteocyte, at which point it just maintains the matrix and doesn't make new matrix anymore, right? Osteoblasts, their job is to make new matrix.
This is happening in the periosteum, correct?
Under the periosteum and also the endosteum as well, but more under the periosteum, yes. That red line that I drew is supposed to represent the periosteum. That is correct. This is all happening under the periosteum. Yep. All right. Other questions on this? All right, excellent. Now, there is something interesting that happens to those osteocytes when they mature. Because they are locked inside of that lacuna, what ends up happening is that they are now surrounded by matrix. Now, that matrix does have water, does have nutrients and things like that, but not quite as much, not necessarily enough to be able to maintain this osteocyte. So what these osteocytes do as they're forming the matrices around them is they form these long processes which allow the cells to interconnect to each other. So what happens is we may have this long, narrow, thin actually, hold on, let's do that again. this long, thin osteocyte over here and a long, thin osteocyte over here. And what these osteocytes have are these long processes that stick off of them. They extend these big, huge, long processes out from each other, reaching out to their neighbors. And so what can happen is their neighbors can end up reaching out to them as well. And when they reach out to them at that location where they meet, they will actually form gap junctions. Remember, gap junctions are those fluid-filled holes between the cells. So maybe what happens is this cell right here on the right is very close to a blood vessel. So it is getting a lot of oxygen. It is getting a lot of nutrients. And so what it can do is it can spread that oxygen and the nutrients to the cells that are farther away. And the cells that are further away can take their waste, can take their carbon dioxide, and they can spread it to this cell so that the cells can get rid of them. So these big, long processes that come off the osteocytes allow them to interconnect with each other and allow them to share resources and move waste more easily to allow the cells to be able to survive locked inside that rigid crystalline matrix. All right. So we have these, and again, here's a better picture. These are the pictures from your textbook, and this one still seems kind of chunky, but the osteocytes become these thin, narrow cells. As we've seen, those lacunas are thin, narrow spaces with these long processes radiating off of them. And the function of these osteocytes are to maintain the tissue. But again, remember, as we mentioned, if released from the lacuna, like from damage or a breaking of the bone, they will revert to osteoblasts and can start making new matrix again. All right? Notice, if you think about it, we said there are 4 cell types inside of our bone connective tissue. But really, if you think about it, they're really just one cell at different stages of its life. It starts as a unipotent stem cell, the osteogenic cell. From there, it becomes the immature osteoblast, and from there, it matures into the osteocyte. So really, it's really the same cell in 3 different stages of its life, as a unipotent stem cell, as an immature cell, and as a mature cell. So when we say there's 4 cells, there's really 4 types of cells. All right, questions on that? Of course, we did say 4, and so we do have that 4th, and one of those things is definitely not like the others. So let's take a look at that 4th cell. The 4th cell type may be similar in name, osteoclast versus osteoblast, but it is a very different type of cell. This cell is actually closer to a white blood cell than it is to actually a bone cell. And again, remember we said the B in blast equals build. Well, then the C in clast, I guess, equals, I don't know, chew, something like that. Because the job of the osteoclasts is to break down bone matrix. Right? Our osteoblasts build matrix, new matrix, and our osteoclasts break down old bone matrix. Now, these are mostly found within the inner lining of the endosteum, although there will be some outside under the periosteum as well. And notice if you look at the anatomy of these, they are very, very different, very, very unusual. One of the things that you notice about it, hopefully right away, is that it has multiple nuclei. What might the advantage of having multiple nuclei be? Remind me again what's stored in the nuclei? What's stored in nuclei?
DNA information?
Yeah, DNA, absolutely. So this cell has a massive amount of DNA. Well, what do we use DNA for? What is DNA the instructions for making? Come on, you guys, it was one of your major physiological processes. Right, polypeptides, proteins, absolutely. We have multiple nuclei because this cell produces a massive amount of proteins, right? Right, the DNA is in there for transcription so that we can then translate it to make sequences of amino acids. Those polypeptides go to the Golgi apparatus, made into functional proteins. This produces massive amount of proteins, and in particular, these are hydrolytic enzymes, right? Their job is to use water to break bonds. And in particular, what they're going to do is these hydrolytic enzymes are used to break down the bone matrix, right? That bone matrix, remember, was the minerals that were pulled out of the interstitial fluid. So when we break that matrix down, it releases calcium and the other minerals into the interstitial fluid. And of course, when we get the calcium and pull it out of the interstitial fluid, when we put calcium into the interstitial fluid completely interchanges with the plasma of our blood. So when we release this into the interstitial fluid, from there, it is going to go into the blood, right? When we get the calcium to make new matrix, it comes out of the blood. So in this way, our osteoclasts break down the bone matrix, releasing the calcium, releasing those other minerals into our blood. And notice that leads us to the other characteristic. Notice it has this massively invaginated surface to it, or what we call the ruffled border. And of course, that ruffled border, as you can see, gives it a massive surface area. Oops. And that massive surface area gives it more contact with the bone for better breakdown of the matrix. All right. So there you go. These are our osteoclasts. Let me see if I can sneak this over in the corner and still make it work. Multinucleated with that ruffled border, and their job is to break down that bone and release it into the blood. And that process of breaking down the bone and releasing it into the blood is a fancy process we call resorption. This is important because it helps us to regulate calcium levels in our blood. And as we talked about, calcium makes cells do wonky things. It is vitally, vitally important that we keep very, very tight and close restraints on how much calcium is in our blood so that our body and its cells can function properly.
Quick question.
Yes.
You said that it has a ruffled border, which gives us the massive surface area. And what was the advantage of that?
Well, because remember, if it's going to be laying against the matrix of the bone, the more plasma membrane it has in contact with the bone, the easier it's going to be for it to release its digestive enzymes and effectively break the bone down. Right? Think of it this way. When you wash your dishes with your sponge, do you use the narrow end of it or do you use the flat end of it? Use the flat end of it because you get more surface in contact with the dishes and you have more of an effect. And so by having an increased surface area, it's more in contact with the bone and it has a larger effect.
Thank you.
Yep. Great question. Any others? All right, excellent. Then just that easily, we have identified the four. So I would say, I'm not sure I would consider them separate functions. Its job is, actually, I will, so let's do that, yes. Its job is to break down bone matrix. One of the reasons we might break down bone matrix is so that we're remodeling the bone or again, as you grow, we need to do the bone or things like that. But when it breaks down the matrix, it is releasing the calcium into the blood. So I think it's like you said, I think the two processes basically go hand in hand. Its job is to break down the bone matrix for whatever reason, whether it's because we're growing, whether it's because we're remodeling the bone, or whether because we don't have enough calcium in our diet. So why it does it may change the reasons, but when it breaks down the matrix, it releases that calcium into the blood. So I think you're right. I would say that they go hand in hand. That process of resorption is breaking down calcium out of the matrix of bone to release it into the blood. Great questions. Love it when you guys ask good questions. That's awesome. Excellent, excellent. All right. Last little bit we need to talk about is we've talked about the cells. Let's see, where are we time-wise? We're doing good. Let's talk about the cells. Let's talk about the matrix of it, because again, it is a connective tissue. It does have cells and matrix. And again, not surprisingly, or maybe surprisingly, because again, normally when we see the bones, right, we just see them as that skull or as that, you know, that bone or whatever it is. Bones are 25% water. There is a lot of water within the matrix, right? It also, and I think we talked about this when we were talking about collagen. Collagen, it is 25% proteins and primarily collagen fibers. This is what makes those bones a little bit flexible, gives the bones a little bit more give to them. So they can resist shearing and tearing forces. So you can twist and turn, you can jump, and you can move your body. You can pound that treadmill as you're running on it or whatever, the ground. And again, they aren't brittle. It gives them strength without being brittle. However, as we also talked about, 50% of it is that crystalline mineralized salt, that hydroxyapatite crystals. Again, I mentioned it to you in the last section. I told you you didn't have to know the term then, but you do need to know it now. This alphabet soup term, which again, you should always expect to have to spell on an exam. Hydroxyapatite crystals, as we talked about, are the strongest organic substance that is made by our body. It is made primarily of calcium phosphate, but also calcium carbonate, magnesium, some other micro minerals are in there as well. As we also talked about, this is the same material that forms the enamel of your teeth. The difference between the enamel of your teeth and your bones is the enamel of your teeth does not have the collagen fibers. That is why the enamel of your teeth does not have any flexibility or give. It is incredibly, incredibly strong, but it has no flexibility. It has no give. So as I mentioned, so if you're eating that clam chowder and you bite on a clamshell, right, you could chip that enamel on your tooth as a result of that. All right. Excellent. Now, as we also talked about, our skeletons don't come fully calcified. I made the joke earlier in the class how you can hold a baby at chest height, drop them, and they will bounce in the ground and come back into your hands. And again, I will say it again, continue to say it, especially because we're not in the classroom. Maybe it's not as easy to tell that I'm joking. Don't drop babies. All right. But as we also talked about, there are plenty of babies that roll off of changing tables and are not injured. And one of the reasons for that is that their bones are not fully calcified at birth. Our bones are not fully calcified at birth. They continue to calcify, or we can think of them as be mineralized as we age. Also, as we continue to age beyond that, one of the things that happens is our collagen fibers start to break down, which is why, you know, babies rolling off changing tables don't get hurt. But if grandma rolled off the changing table and fell to the floor, right, grandma shatters, right, because her bones have less collagen in them, so they become more brittle. All right. So again, our osteoblasts are depositing those mineral salts between and on the collagen fibers like that rock candy. And that is that process of ossification or calcification or mineralization, whatever you want to call it. It is the process of how we make bone matrix. All right. Now, we use this process of ossification to make two different types of bone tissue. So remember when we talked about bone tissue, we said that there are two main types, compact and spongy or cancellous. For the previous test, we just needed to know compact, but now that we're on the skeletal system, we will talk about both types. All right. Again, this is where we get very vocabulary dense. So even though it's probably a little bit early for a break, this is probably a good natural stopping point. So let's go ahead and take our first break now. We'll take our first break of 15 minutes, come back in 15 minutes. So we will restart at 9:15, and I will start the recording at that point. So any questions before we take our break?
Can you leave this slide up?
Yep, sure, I can do that. Although again, I'm happy to leave the slide up, but I will also remind you, remember, you have all of the slides in the modules on Canvas. So you do have those as well. So you don't feel you have to write all these things down because you do have it that way. But if, again, writing this is how you learn, then by all means do that. All right, any other questions? All right, I will see you guys in 15 minutes. Let's go ahead and get started. Any questions before we dive into new material? All right, then let's go ahead and dive into new material. As we mentioned, this process of calcification, this process of ossification forms two main types of bone. The two main types of bone, as we talked about, are compact bone and cancellous or spongy bone. Compact bone is by far the most common type of bone connective tissue. Making up something like 80% of the skeleton. It is also the superficial bone type. So when you, again, assuming we were in the classroom, if we were to pull the skeleton out and look at good old Bob there, you would see that all of the bone that you saw would be compact bone. Now, again, one of the main ways we are going to identify our types of bone is by their organizing structures. And as we've already seen with compact bone, their organizing structure is that osteon, those concentric circles around that central canal. And again, we'll talk about all the anatomy of that in just a minute. Making up about 20% of your skeleton is our cancellous or spongy bone. And again, both of these are appropriate anatomical terms. So if spongy is easier to spell, then by all means use spongy. If you like the fact that cancellous sounds more impressive, then use the term cancellous. Whatever makes you happy, both are appropriate anatomical terms. But it gets the name spongy because of the way it appears. If you were to go down to your kitchen right now and look at your kitchen sponge, what you'd notice about the kitchen sponge is it has all these irregular points and processes. It isn't just a single repetitive pattern. It isn't like a honeycomb where you see the same shape over and over and over again. It looks very irregular, and so that's actually how we describe it. This trabeculae is basically an irregular point or process. Right? So where is that osteon? And I'll just cheat and draw the circles, even though they won't be as pretty. As we've seen, it is a bunch of concentric circles around a central canal. That is what our osteon looks like. If you look at the edge of the sponge, at the edge of the sponge, the tissue is going to be made up of all these things like this and things like that and things like this. So again, each single one of these irregularly shaped point and process is known as a trabecula. Trabecula singular, trabeculae with the E is of course the plural, right? Osteons plural, trabeculae plural. So here we can do that too. Fun with vocabulary. All right, and again, I've done a horrible job of drawing this, but let's look at the pretty picture to see how this works. And again, I already mentioned it, but I'll say it again. About 80% of the skeleton is compact bone, and only about 20 is spongy. And again, compact bone is all the superficial bone. Bone is compact bone. So when you look at a skeleton, unless the skeleton is damaged, you only see compact bone. All right, let's again, instead of me having to draw them, let's look at the pretty pictures of this. And we're actually going to start first with an illustration from your textbook. We see a little of the anatomy that we've been talking about. Where's my highlighter? There it is. So notice here we have a long bone that we've cut out a little chunk of and are looking at. Notice it shows us some of the features we need. For instance, that dense irregular connective tissue on the outer surface, what we call the periosteum, is there. Notice we said the blood vessels can be stabilized from it. Notice also, as they're pulling it off on this side, it doesn't pull off easily. The reason for this, and I'll cheat and I'll draw some lines, is remember we said this is a dense irregular connective tissue, and as we know, dense irregular connective tissues have a lot of collagen fibers in them. Well, one of the things that happens is those collagen fibers from the periosteum penetrate deep into the bone matrix, really anchoring it and holding it in place. This helps to really attach that periosteum on the outer surface. Remember also we said how this is also where our tendons and ligaments attached. And remember I told you a tendon is actually strong enough where if you pull on that tendon, you are more likely to break the bone than you are to tear the tendon. And that's because the collagen fibers from the tendon will also penetrate into the bone, anchoring it in place. Right? These fibers that extend deep into the bone out of the periosteum are what are known as the Sharpie fibers, because good old Bob Sharpie was the first person who identified them, or because they perforate into the bone, they're also known as perforating fibers. And of course, I'm sure you have your study guides, both histology and the gross anatomy in front of you. And sure enough, you would see that those Sharpie fibers or perforating fibers are one of the things that you're responsible for histologically, like on an illustration like this. Notice as we look closely, we can see plenty of examples of osteons in the compact bone. Notice with those blood vessels and nerves at the center of them. Here, some of the layers of matrix have been pulled out for us so that we can see these. But notice on the inside of our long bone, we have all these irregular points and processes. Notice there isn't just one repetitive pattern, right? This. It's a repetitive pattern we see again and again and again. But not here. Here we have all these irregular points and process that make this cancellous bone really look like a sponge. And so every single one of these, this long one, this short one, this branching one, all of these irregular shape points and processes would all be referred to as trabeculae. So these trabeculae form the spongy bone, and these osteons form the compact bone. So this is the organizing structure of compact bone. These are the organizing structures of our spongy bone. Now, if we were to, like they did in the picture, pull, one of the osteons out, what we see, as are mentioned, is layers of matrix. And again, because vocabulary is important, this layer of matrix that makes up an osteon is called a lamella, lamellae being the plural. However, these specific, so this is a general term, a lamella is a layer of matrix. However, if you notice, to make an osteon, we have concentric circles of lamellae that wrap around each other, and that's actually what they're called, concentric lamellae. Oops. Lamellae being the plural of that, and they are used to form the osteons. Notice what's nice about this illustration is you can see, and let me switch back to my highlighter, that again, we know that you make the matrix by wrapping it around collagen fibers. And notice that the collagen fibers wrap around at different orientations within the different concentric lamellae. And again, as the illustration shows, this gives it resistance from shearing and bending and twisting forces in no matter what direction you use it. Now, basically, it's a big straw. And one straw by itself doesn't have a lot of strength. But if you have a whole huge bundle of straws in your hand, then again, you have some flexibility and give, but it's going to be very hard to break that in half. And that's essentially how compact bone works. by having these resistance straw-like structures, they can resist shearing and tearing forces and provide a tremendous amount of strength. All right, so here we have, again, as we mentioned, that example of the concentric. Here, we actually see it in the light microscopy. And so again, I'll use my highlighter for this one first. That color will work. So again, we have these layers of matrix that are going around, and that layer of matrix that goes around is a lamella, but again, not just any lamella, but as we talked about, a concentric lamella, because they are forming those circular patterns that form an osteon. All right. Now, we didn't mention this when we looked at it last time, but notice as we look closely, remember we know this space here is our lacuna. And as we talked about for the last section, we know these lacunas contain the osteocytes. But now we know something new about the osteocyte. Remember, we learned that the osteocytes have these long processes that stick out that allow it to communicate to the cells next to them. Well, if you're going to wear a shirt and you have arms, there have to be holes in the arms of the shirt for your arms to pass through. And notice if we look closely at our light microscopy, we see that. Lo and behold, here we have a lacuna, and there are all these dark lines that are radiating out of it. These dark lines are the sleeves for the processes. So you can see how the process of one would connect to the process of the other. Here are some others that are really prominent, really distinct. We can see these long, tiny, in fact, they're like, oh, here's some really good ones. Some really, really long, narrow lines, dark lines that are radiating out of these lacunas. Those spaces, remember the lacuna is a space, so these tiny little things are spaces as well. They're tiny little canals that the processes of the osteocytes reach out with, and in fact, that's their name. Canaliculi means tiny canal. So the lacunas all have canaliculi that come off of them for the processes of the osteocytes to radiate out. All right. Questions on that? All right, let's talk some more fun with vocabulary. Looking again at this nice illustration from your textbook. Again, an illustration of the histology is definitely something you could be responsible for on the exam, as are some models, and I will show you some examples of that in just a minute. But where are we? Oh, yes. So, as I mentioned, here we see again those osteons forming our compact bone. And again, with my highlighter, we can see that there are these circular layers of matrix that are forming the osteons. As we mentioned, they are what we call our concentric lamellae. However, notice on the outer surface of the bone, the layers of matrix aren't forming osteons. Instead, they're almost like a straight line, but if you think about it, if we extended out this chunk of bone, it would go around the entire circumference of the bone. And so these relatively flat layers of matrix that go around the entire circumference of the bone are what are known as circumferential lamellae. Lastly, and I'll change the color even though I didn't change it for the last one. Notice there are some, and again, it needs to be a solid mass. And if you think of having a bundle of straws in your hand, there is going to be some space in between the straws. We don't want space in between the straws here. So notice there are some irregular pieces of bone, some irregular shaped layers of matrix. Some are very long and bendy, some are very short and squat. But there are these irregular layers of bone, and these irregular layers of bone are what are known as the interstitial lamellae. So if you notice, when we look at compact bone contains all three types of lamellae. concentric that form the osteons, circumferential that go around the entire circumference of the bone, and the interstitial ones that are located within the, between the osteons is probably the best way. It's within the tissue, interstitial, within the tissue between the osteons is probably the best way to describe it. All right.
Question?
Yes.
I'm having a little bit trouble understanding the difference between the concentric and the circumferential.
Okay, so let's change colors. Notice this is a pie wedge piece of a bone, right? So if instead we made an entire circumference of the, whoops, wrong button. Hold on. So here is the entire bone. And then in the center, here is where the cavity is at the center. And again, what they've done is they've taken a tiny wedge of the bone like this, and that's what we're looking at, okay? Are we oriented here?
Yeah.
Okay, excellent. Now, as we mentioned, and let's say I used red for it, so let's switch to red. We know that the compact bone is comprised of osteons. So here's an osteon, and here's an osteon, and here's an osteon, and so on and so forth. And each one of these osteons is made up of a circular layer of matrix that surrounds another circular layer of matrix, which surrounds another circular layer of matrix until we finally get to the central canal. All right?
Okay.
So what I've colored here in red is the circumferential lamellae. Pardon me, the concentric lamellae. All right? So those are the concentric lamellae. They're concentric circles. That's what concentric circles mean. They're circles inside of each other. Right? Now, what's out here, and I'll change the color to blue. This layer out here looks relatively straight because what happens is it goes around the entire circumference of the bone. We're just seeing the little bit of it that is right here in the pie wedge, but it actually goes around the entire circumference of the entire bone. And that's why it is called the circumferential lamellae, because it goes around the circumference of the circle, around the entire structure of the bone. Whereas the concentric circles just form the osteons.
Oh, gotcha. Okay.
Okay. Changing the color, did that help? Help to make sense?
Yeah.
Okay, perfect, excellent. Great question. Any others? Because again, I appreciate this is dense. I appreciate there's a lot of vocabulary on this. So it is important that we not get confused. So if anybody else is confused, like I said, please, by all means, ask these questions because that's what I want you to know. All right, so to keep, yes, go ahead.
They're still the same function, correct?
They're still layers of matrix, so their job is still to make the bone, absolutely. So they're just layers of the hydroxyapatite crystals and collagen. They're forming the bone. They just have a different shape to them, so that's why we give them different names.
Same thing with the interstitial, correct?
Yep, all
That's
three.
the
All
shape.
three are just different shaped layers of matrix.
All right.
But you're right, a layer of matrix basically serves the exact same function, helping to provide the strength and integrity to the bone. But so now if you think about it, now we know 3 things about compact bone. Well, 4. It's the most common, making up 80% of the skeleton. It's superficial. It's towards the surface of our bones. We know its organizing structure is an osteon. And now we know that it is comprised of all 3 types of layers of lamellae: circumferential, concentric, and interstitial. Okay? This is important because it helps us to distinguish it from our spongy bone. We know there's less spongy bone, only making up 20%. We know the spongy bone is deep. So the spongy bone, notice, and again, we'll go with green, right? This stuff here is the spongy bone. And we know the spongy bone now is going to be located here on the inside of our bone. We know it is made up of irregular points and processes, and these irregular points and processes are known as the trabeculae. But notice they've conveniently cut one of these open for us. And when they cut it open, we can see that it is comprised of layers of matrix. So it's still going to be layers of matrix that form our trabeculae, that form our spongy bone. But it's not going to be all 3 types like our compact bone. So we did that, we did that, we did that. We made sense of that. Ah, excellent. We know the central canals run down the center of the osteons that are like big straws. But notice also that if we just had the blood vessels running parallel within these, they would have to enter at one end of the bone and exit at the other end of the bone. And that's where we form our joints. So that's not very efficient. So notice what happens is the blood vessels that are outside of the bone need to penetrate into the, that color didn't work, brown. Needs to penetrate into the bone and also penetrate between the osteons to get inside of the bone. So notice we have these parallel spaces that are the central canals, and the ones that penetrate or perforate the bone are what are known as the perforating canals. And those are the appropriate terms to use, central canal and perforating canal. There are more antiquated terms, Haversian and Volkmann canals, again named after the two dudes who described it. Those are stupid terms. They're very antiquated or not used anymore. So I would prefer, again, I won't mark you wrong if you use them on the exam, but it is now much more appropriate to use central canal and perforating canal for these spaces. Yes?
Could you please, what was the difference between the two? I didn't.
The orientation of them. Central canals run parallel, they're at the center of the osteons. So basically they run parallel to the osteons.
Right.
Whereas the perforating canals are the ones that are perpendicular to it.
And that would just, because in the picture it looks like it's the same set of canals. But is it just that if you point towards them, that's when we're supposed to tell, okay.
Yeah, so on the exam, if I asked you to identify, well, that's a horrible shape. If I pointed to this on the exam and asked you to identify that space, you would say?
Central.
Right, but if instead I pointed to this space, you would say?
Perforating.
Perforating. And again, what I will say, and this is probably a good point, a good place to bring this up, it's a teeny bit confusing there because it has the blood vessels in it. Now, if we were in the classroom, one of the things that I would be able to do is pull out one of our models for us. And we'd be able to look at that. Conveniently enough, I just during the break posted these pictures onto our Canvas site. So let's share this. Excellent. So notice here, this handout, can you guys see this?
Yes.
Yeah, excellent. Okay, perfect. All right. So this is a model that is in the classroom. So if you look, this handout is on there and there are multiple pictures of it. Hold on, I gotta get all my ducks in a row. Here we see a side angle, whoops. Here we see the other side. Here we see it from the front or the back, I guess, depending on how you think of it. Here we see from the back or the front, depending on how you think of it. And here we see it from the top. Notice this is kind of the view that we were just looking at on the previous picture. We see our, and let's go ahead and use my highlighter again. We see our, green, that doesn't show up well. Let's try green. We see our concentric lamellae that are forming the osteons. We see our circumferential lamellae. In fact, notice they've pulled some of them out. They almost look straight in their structure, going around the entire circumference of them. And notice, and we'll use blue, there are some areas of just irregular appearing layers of matrix that fill in the gaps kind of in between. So here's a little gap filling, here's a little gap filling, our interstitial lamellae. Notice they've also even, and I'll switch to my highlighter here, have emphasized the lacunas, and we can see the little lines radiating out from them, showing us our canaliculi on there. They show us the blood vessel in the central canal as we look at it there. And as we go back to the first view, right, notice here we have the blood vessels in place. Ooh, notice they do a great job of showing us our Sharpie fibers here, those extensions of the collagen fibers, those perforating fibers. So here we show the blood vessels that would be in the central canal and the perforating canal, but conveniently here, they've pulled the pesky blood vessels out. So notice these blood vessels that are coming through, and notice also on your illustration, they're perfectly perpendicular. They're not going to be perfectly perpendicular, but these that are either going deeper or more superficial, this would be the perforating canal. And the ones that run parallel to the osteons would be the central canals. So this is a central canal, and this is a central canal, and that's a little bit of a central canal, and this is a central canal, and this is a central canal, and this is a central canal. So we can see all the central canals there. And we can also see the perforating. This would be perforating, this would be perforating, this would be perforating, 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 and so on and so forth. Does that make sense? Silence, I'm
yes,
going to assume means yes. Excellent. All right, perfect. Notice two other things. Notice that as we looked at the end views, again, here we see that periosteum on the outer surface of the bone. And unlike the picture in your textbook, here in blue, we see the endosteum as well. So notice what they've shown us here is the endosteum, that dense irregular connective tissue on the inside. But notice they've removed it in some of the places so we can actually see the spongy bone in between. So here we have the spongy bone in between, those irregular trabeculae. This one's short, this one's curled, this one's long and winding, this one's narrow, this one's thicker. Again, there's not just one form to a trabecula. There are all of these irregular points and processes. And we see that really nicely on this model.
Can we zoom into this picture one more time?
I'm sorry?
Can we zoom into this picture?
When you're looking at it on...
Oh, right, you said posted it? Gotcha.
Yeah, I've posted these, and so again, you should be able to increase the magnification of this as you go. It's a PDF that's on there. The other point that I would like to point out, and I'll be honest, that like one of those things you're never supposed to ask a question you don't know the answer to, but let's take a look at this and see if I can find something real quick. Yeah, it's not here. Bummer. Oh, wait, no, maybe. Ah, it is. Sweet. Excellent. All right. Where are you guys? There you are. So, if you will recall, one of the, again, if you go in the course modules, under the course modules, under the study guide, the first link is to Cosumnes River College's Virtual Anatomy Lab. If you click on that and come to this page, they have a lot of great resources that are going to be incredibly helpful for you. Because they have, for instance, as we're going to go starting to do today, learn about the axial skeleton. They have pictures of bones and models like this, and then notice they have unlabeled ones. And if you click to the next slide, it actually shows you characteristics and features. It gives you the labels for these things as well. It is incredibly useful that way. Notice the other thing they have here is histology. If we look at histology, notice they show us here a histology of some compact bone. We can see the osteons, we can see those things, and they have the labels for them as well. Notice when you're looking at the bone this way, notice our central canal is at the center, and you can actually see the perforating canals as it's traveling from one osteon to another. So there's another perforating canal that we can see as well. Some more anatomy, but the other thing they have the exact same models that we have. So notice they have a picture here, and then they have labels for that you can see as well. So again, not only have I provided you with the pictures, but you also have this tool from your study materials from Cosumnes River that actually does an excellent job of showing all of these things as well. So we can see those really, really nicely, and it has labels. Again, if you're not using the resources that are available to you online, shame on you. Make sure you're taking advantage of these. And again, you just get right to that link from under the study tools, the very first one, Cosumnes River College Virtual Anatomy Lab. As I mentioned, I just posted this. In fact, here, I got to refresh the page so we can see that. There we go. Bone model pictures are those pictures. So those are posted now under the lecture images at the bottom. All right. So Great resource. Make sure you are taking advantage of it.
I had two questions.
Yes.
The first one was, you said that Trabeculae also has what kind of lamella?
Lamellae? I haven't said yet, but that's exactly where we're going next. That is the perfect question, because that is exactly where you're going next, because you are absolutely correct. What I mentioned is that we can see that it's made of layers of matrix, but I didn't say which one. But let's think about it though, okay? So remember, and there's my highlighter. There are three types of lamellae. Circumferential go around the entire circumference of the bone. These are deep inside the bone. So is there any way that these could be circumferential lamellae? If you're in the inside of a house, is there any way you can paint the outside? No, there's no way you could do that. So that's not the case. Notice concentric form perfect circles around other perfect circles around a central canal. They form the osteons. Are there any osteons in spongy bone? No. So it can't be circumferential. It can't be concentric. So as it turns out, the trabeculae, and again, I think I'm going to say this on the next slide, but we'll do it since this was a great question. I'll go ahead and answer it now. The trabeculae are formed by only one type of lamellae. And what is the only type of lamellae that can form the spongy bone, that can form our trabeculae?
Interstitial.
Interstitial. Perfect. Trabeculae are only formed by interstitial lamellae. Irregular shaped layers of matrix make irregular shaped trabeculae. So absolutely perfect question. And that's exactly, like I said, where we're going to go next. But I love that you asked it. So here we go again. Notice here we see that spongy bone on the inside, much more of it in the heads of our long bones, but still even our bodies of our long bones, the shaft of our long bones have a little bit lining the inner surface. They're made up of all these irregular points and processes with our endosteum on the surface, but notice inside of it, we have those irregular shaped layers of matrix. Notice we still have lacunas, we still have canaliculi, we still have layers of matrix, but our trabeculae are, and again, I'll write it again, only formed by interstitial lamellae. All right, trabeculae, let's say it this way. Trabeculae, okay, which of course form the spongy bone, are only formed by interstitial, oops, that's way rough, interstitial lamellae. All right, excellent. And notice they've peeled off the endosteum on this one, but if you notice, they actually left the immature cells. So here we have the osteoblasts that are lining up, the osteogenic cells, and notice they even gave us an osteoclast with its ruffled border and its multiple nuclei, because we know there are more of these osteoclasts inside the endosteum. So even though they peeled off the dense irregular connective tissue, they left all the immature cells there for us.
Quick question.
Yes.
In the book, there was a picture of when of a white blood cell engulfing another, I believe, a bacteria, and I looked at it and I'm just thinking, is it the same thing that's happening here? Is it the same process where the osteoclast just engulfs the entire?
No. So no, in this case, remember with that large surface area, what it is doing is it releases digestive enzymes via exocytosis. And those digestive enzymes break down, dissolve the matrix, and release it into the interstitial fluid.
This is the osteoclast, right?
Yes, the osteoclast releases digestive enzymes that break down the matrix. So notice this matrix was originally, a continuous layer. And what it's done is it has eaten away at it by using its digestive enzymes to break down that matrix. And now there's more being released into the blood. Next. All right. Excellent. Excellent questions. Any others?
Quick question.
Yes.
The osteoclast do they like multiply, or is there more of them as you get older?
So there isn't necessarily, so first of all, remember we said the only bone cell that is capable of dividing is the osteogenic cell. And osteogenic cells don't make osteoclasts. Remember, osteoclasts, I said, we're closer to white blood cells. So if we need more of them, our mesenchymal cells will actually make more osteoclasts, okay? What happens, the reason our bone density decreases as we age isn't so much that we get more osteoclasts. It really has more to do with the metabolism of the cells, right? If you think about you as a healthy adult right now, you have osteoblasts that are constantly making bone matrix, and osteoblasts that are constantly making bone matrix, and osteoclasts that are constantly breaking it down. And if your job as a healthy adult organism is to maintain homeostasis, what should the two rates of those processes be?
1 to 1?
Yeah, 1 to 1. So the same rate that you're making new bone matrix, you should be breaking down the matrix, and so you maintain level amounts of bone matrix. The problem that happens as we age, our osteoblasts become less active. So you keep breaking down bone at the same rate that you were before, but now you're not making bone matrix nearly as quickly. And so what happens is our bone density starts to decrease, right? But what about your 3-year-old that's running around the room? Are they making bone and breaking down bone at the same rate?
No, they're making it faster.
Yeah. When you're younger, as we're those growing adolescents, we make bone much more rapidly than we break it down. So when we're immature, we're building up our bone matrix, then we maintain balance, and then as we age, we slowly start to lose that density of bone. All right?
Okay.
Excellent.
Are we going to be, is there a difference between the external and internal circumferential lamellae, or no? Difference in function?
Well, again, there's only external circumferential, right? Because remember, circumferential are only on the outer surface.
Gotcha. I'm sorry, just the picture I was looking at had 2 different ones, so I wondered.
Well, so let's go, I think, hold on, oops, wrong direction. So again, there are multiple layers of concentric lamellae, but really, if you think about it, lamellae is just a layer of matrix, right? It's a piece of paper. The only difference between the circumferential lamellae and the concentric lamellae is the shape of this. This piece of paper is the same, whether I hold it flat, whether I curve it, or whether I crump it up into a ball, it is still the same layer, it's still the same paper. The only difference really is its shape. And that's what we're talking about here. When it is flat on the outer surface of the bone, it's a circumferential lamellae. When it's in a circle, and I can't make a good circle out of this now because I've crumpled it up. When it's in a circle, forming those osteons, again, it's the same layer, it just has a different shape to it. And then if I fold it in half like this, it's still a piece of paper. So really, the only difference between the circumferential, the interstitial, and the concentric is their shape. And then the only other difference is where they're found. Circumferential is only on the outer surface of the bone. Concentric are only in the osteons. And compact bone uses all 3 types, whereas spongy just uses interstitial. But yeah, a layer of matrix is a layer of matrix is a layer of matrix. Its job is the same no matter what its shape is. The only difference is its shape and where we find it. All right? Questions on that? All right, excellent. With that, then, I think we've hit most of the microscopic anatomy. Here, let's cheat. Go back to there and look at our histology. Whoops, what's going on? That's weird. All right, hold on. I'm gonna stop sharing for a second. I don't know if you guys saw that flickering, but I did, and it was weird. So let's try this instead. There we go. That's still kind of doing it. Oh, that's odd. All right, so hopefully you've already downloaded your histology. Osteon, osteocyte, concentric interstitial, endosteum, perforating lacuna. Looks like we've hit all of those things. I guess the only thing we didn't talk about is bone marrow. Let's talk about that in a second. Trabeculae, interstitial, lacuna, endosteum, osteocyte, red bone marrow. Okay, perfect. So that's really the only thing we have left to talk about. So for that, then let's go back to our slide. As we talk about our, and we'll come back to this slide and bring back my annotation tool. As we will talk about, as we're about to talk about in our bones, remember the picture, actually I lied, let's go back couple more pictures to this one. Wait, where'd that picture go? That's the one I want. Excellent. All right. Last little bit of information that we need to add to this is to talk about the bone marrow. Remember, as we talked about as an adolescent, all of our bone marrow in our body, as a baby, as an infant, as an adolescent growing, our bone marrow is all red bone marrow. However, as we start to age, our bone marrow starts to accumulate adipose in it. And as it accumulates adipose in it, it fills up with adipose and it becomes yellow bone marrow. That yellow bone marrow is in this big, deep hole, this cavity at the center of our long bones, known as the medullary cavity. So as a mature adult, all of the marrow in this space, which you think about it would be this space here, or at the end of that bone picture that we saw, is going to be the yellow bone marrow. Notice our spongy bone is primarily in the heads of our long bones. And even as mature adults, we still need to be able to make new red blood cells and new white blood cells and new platelets. So up here in the spongy bone of the epiphyses of our long bones, and then within our flat bones and other bones, is where we're going to find our red bone marrow. So within these bones, there is a distribution of our bone marrow types. Spongy bone tends to have the red bone marrow filling these spaces, whereas the big space within our medullary cavity is going to be where we find our yellow bone marrow. So those are the types of bone marrow that we have, yellow and red, and the locations of them. Other than that, it looks like we covered everything on our histology handout. Notice there's not nearly as much histology for this section as there was for the last, but there's a lot more gross anatomy. All right, excellent. All right, so then with that, let's talk about the 6 structural classifications of our bones.
Can I ask a quick question? Sorry.
Yes.
You said the spongy bone will, once it starts to change the red marrow into the yellow bone marrow, that's where it'll house the yellow bone marrow or the red one?
Red. The heads of the bones, the spongy bone will always have red bone marrow typically in it.
And yellow will be?
Within the shafts of the long bones.
Got it. Okay, thank you.
Yep. All righty. So your book does a good job of describing all 6 structural classifications, but it does tend to emphasize 4 of them more than the others. So I do want to make sure that we're clear on this. There are indeed 6 structural classifications. Normally when we talk about anything in the body, we talk about their structural and functional classifications. And the good news is there really aren't different functional classifications for bones. So when we talk about the bones that make up your skeleton, there are just structural classifications, and there are 6 of them. Give me an example of a structural classification of one of the bones we see in the body. Just finished mentioning a couple of them. As you look at the skeleton, what are some of the structural classifications? Well, those are the parts, but the diaphysis, so you're right. A diaphysis would be, for instance, this part of this bone, right? The shaft. The diaphysis is the appropriate term for the shaft of a bone. But what type of bone is that? What type of bone am I pointing at there that I've just highlighted? What is the type of bone that actually is? There you go. That's what I'm looking for. Long bone. Long bone is one of our structural classifications. How would you identify a long bone?
By how long it is.
Okay, sure. The building is tall because it is tall, right? So yes, a long bone is long. But we should probably, that's correct, it's not an inaccurate statement, but we should probably be a little bit more descriptive in that. Typically a long bone is a bone that is thin, narrow, right? Elongated. I know that's just another way of saying long. A thin, narrow, and typically is longer than it is wide, right? So it's a thin, narrow, elongated bone.
Are the long bones in our body, is it just the ones in our extremities?
You are correct. That is a great point. All of our long bones, whoops, are found in our extremities, or again, we can be more specific, appendicular skeleton. And in fact, all of the bones of our appendicular skeleton are long bones, with really just two exceptions. What are the two main exceptions between our long bones that are making up our arms and our legs?
The patella?
Okay, so you're right. 3. So the patella is 1. We'll get back to the patella in a second. What are the other 2 exceptions?
Metatarsals and
Well, not the metatarsals, but the
Tarsals.
tarsals. Excellent, the tarsals. And what's the other ones?
carpal.
Carpals and tarsals. Excellent. What type of bones are those? Those aren't long bones. Those are
Short bones?
short bones. Short bones are bones that are small, block-shaped bones. And as was mentioned, they're found in the carpals, basically the base of the hand, what we commonly refer to as the wrist, and the tarsals that make up the heel and posterior part of the foot. So the examples of these are the tarsals and carpals. Excellent.
What is the patella?
That is a great question. What is the patella? Anyone know? It's found in the knee. Absolutely. It's what we commonly think of as the kneecap. Anyone know what the structural classification of the patella is?
Would it be the irregular?
Nope, not a bad guess, although that is one of our characteristics. Regular bones. What's another one?
Sesamoid?
There you go. That's the one for the patella. Perfect.
Is it? Oh.
Yep. Sesamoid. Excellent. We need 2 more. What are the other ones?
Flat.
Flat. Excellent. Perfect. So let's stop there for a second before we get the last one. Oh, we can go ahead. Anybody know the last one?
No.
I mean, I can cheat and look in the book, yeah.
No, what's wrong with cheating? With this part of it's not cheating if we're working together to learn this material. I wouldn't call that cheating.
It'll be the sutural or sutural?
There you go, sutural.
Sutural.
Excellent. Bones. Perfect. Excellent. We have now identified all 6 types. All right, let's start first back up here with the flat bones. How would we define a flat bone?
Ribs. They're flat and narrow.
So again, remember, we want to be careful. Typically with a flat bone, what the flat bones are again, they're going to be thin, but they're unlike the long bones, which are narrow, they are more broad. So thin, broad, right? Again, I hate using the term in the definition. A flat bone is flat, a tall building is tall. That isn't, but we have a thin, broad bone. There's one other characteristic that flat bones tend to have to them. What's the other characteristic that flat bones, like the ribs, like the bones of our skull, have to it?
They're easy to break.
True. They can be, although I would argue that some of these up here can be pretty hard. Curved, that's the magic word I was looking for. They're typically curved bones. So they're broad, thin, curve-shaped bones. Excellent, right? Again, if we, my drawing skills are horrible, but if we think of a long bone is typically what we think of like that dino bone, where it has a thin, narrow shaft and then that elongated, enlarged head to it. Whereas our flat bones are thin bones that are curved. They're narrow, but they're typically much broader, right? So they're broad, flat, narrow bone to them for those flat bones. Short bones are the block-shaped bones, right? So we have all of those. So then what's an irregular bone?
Is it a bone that has two or more characteristics, as you know, like maybe it's curved and also like blocked?
That's not a bad way of thinking of it. You got the right idea. And in this case, we're actually going to use the other terms in it. Basically, the way they define an irregular bone is that it is a bone that is not flat, long, or short. All right? So it's one that doesn't fit any of those other three categories. Remember, I mentioned your book emphasizes 4 of the main types. Although it does define all, oops, wrong way. Not back there. What I want is this one. Let me grab that. All right, back with it. We'll put it back there. Irregular bones, like the vertebrae, are bones that are not flat, not long, not short. The reason your book tends to emphasize these 4 categories is almost all of your named bones fall under one of these 4 main categories. So almost all of your named bones, of the 206 named bones, are either long, short, flat, or irregular. But of course, the key word in that sentence is almost, because almost is not all. There are a couple exceptions. One of them, as was mentioned, is the patella. The patella, as you guys mentioned, was the kneecap. It is a sesamoid bone. Sesamoid bones are actually bones that form in ligaments. Almost everybody has at least 2 of these. And as you guys mentioned, it's the kneecap. Notice again, I said almost everyone. Reach down and feel where your knee is, right? There are people who are missing one, or in some cases, both kneecaps. Most people, and especially in a class this small, it's unlikely that there's anybody who doesn't have one or both of their kneecaps. I always ask, but even usually if there is somebody, they don't want to admit it. But I'll ask anyway. Is there anybody here who doesn't have a kneecap? One or more? And again, removing it surgically doesn't count. Anybody here not born with or didn't develop their patella? Yeah, most people have them, very few people don't, but it does occasionally happen. Those are, because they're so common, are given a name, as we mentioned, the patella, and so we have 2 of those. But these can form in over a dozen different locations, typically areas where the ligaments and tendons are undergoing a lot of stress. So for instance, this can occur within the ligaments of or the tendons, ligaments. Hold on, let's do it this way. Form in tendons. Sorry, I misspoke. Too much things going on. Yeah, form in tendons. Typically tendons that are undergoing a lot of stress. So places like the wrist, places like the ankle, places like the hip. Professional bull riders will actually form sesamoid bones on their inner thigh within the tendons there from the riding of the bulls. So as you put different stresses on your body, what will happen is these bones form within those tendons to try to slightly change the angle of the tendon to make it more efficient in dealing with those stress. Now, there's over a dozen different places in the body where they can form. Does that mean that every single person gets them in the exact same locations inside of every single person's body?
No.
No. And so that's why, and remember, we talked about everybody has far more than 206 bones. We only have 206 named bones, right? Because I may have 3 in this wrist and none in this wrist, right? Or probably vice versa, because I'm left-handed, 3 in this wrist and none in this wrist. 2 in my knees, 1 in my ankle, whereas you might have 4 in your ankles and 2 in your fingers and all sorts of things. Since we have them in different locations, we can't give them meaningful names, but they're just a different type, a structural classification of bones. And the same thing is known for sutural bones. Sutural bones are also sometimes referred to as wormian bones. And these are bones that form between the bones of the skull. What happens is, and I'm going to need to give myself a little bit of room here. So let's get rid of, where's my eraser? this.
Are they the bones that connect each different bone in your skull? Is that what you're talking about?
No. And so, yeah, exactly. No, that is not what they do. And so that's what I want to make sure to emphasize that. So, oops, hold on. Too many moving parts. Hold on, give me 1 second. Okay. So let me draw this because it'll make more sense if I try to draw this. So,
Are they the bones that kind of fuse together when you're like a newborn? They're like spaced apart a little.
sort of. You've got the right idea. Let's take a look at your skull. This is, oh, God. All right, I'm going to cheat and use the straight line because I'm incompetent. There we go. There we go. There we go. And there we go. All right, this is a part of your skull. What happens, as you guys have mentioned, is that we have bone A up here, and we have bone B down here. And as we mature after birth, what will happen is these two bones grow into each other. And they grow into each other irregularly. So typically there's these big, huge undulations to it as it grows into each other, forming the joint between the bones of the skull that we call a suture. So these joints that form the bones of the skull together are what are known as a suture. They're these irregular knit together bones. They're a joint where they come together, making it immovable. But notice you can clearly tell what is bone A. So right here, is this bone A or bone B?
Bone A,
Is this bone A or bone B?
B.
Right, and so on and so forth. However, what will actually happen is as a suture forms, small little bones will form within the sutures. Most of these are about the size and shape of a grain of rice. So they're really, really small. However, some of them can be as large as a $50. So, and again, these form irregularly. Not everybody has them in the same locations. Right? So this clearly is a separate bone. It is not a part of bone A, it is not a part of bone B. It is a completely separate bone that has formed within that suture, and so it is called the sutural bones. And does everybody have the same number of sutural bones in the exact same locations? No. So again, sutural bones, most of which are the size of a grain of rice. Sesamoid bones are mostly the size and shape of a sesame seed. That's where they get their name. And again, but you can have dozens of different locations where they found, dozens of different ones. When you put them all together, everybody has far, far more than 206 bones in their body, but only 206 of them are named because the sutural bones and most sesamoid bones do not have names because they're irregular in where they form.
Question.
Yes.
So the sutural bone, does it always connect like two bones together, like almost like a matrix or like the glue between the two bones?
But it may serve that function, but if you think about it, if it's the size and shape of a grain of rice, is it really doing a whole lot to hold those two bones together?
No.
Probably not. So they're just a physical characteristic that occurs. Some of them can be large, but most of them are pretty tiny.
What is their purpose?
Who says they have a purpose?
Everything
they're
in the
just-
body usually has a purpose, right?
I would say that is not always entirely the case, that sometimes things just occur. Yeah, what you got to remember, the goal of life is to be efficient, right? But not to be perfect. Life is lazy. Once it figures out how something works, it's going to use that process again and again and again. And even if there's something more efficient, if this is enough, it's going to be satisfied with that. Maybe just the way that it comes together, the additional bone forms in these spaces. Why is always one of those questions that's really hard to answer. We don't know why these form. We just know that they consistently form, and they consistently form in different locations. So why that occurs isn't fully understood.
Okay, so you can have bone A and B and not have a suture in the middle, correct?
No, you would definitely have to have a suture between them, but you won't necessarily have a sutural bone.
Gotcha. Okay.
Okay. Again, let me get rid of my pretty pictures. My pictures, and let's look at the pretty pictures from your textbook. Right. Here's our broad, thin, curved, flat bone. There's our long, thin, narrow, long bone. Here are our block-shaped short bones. There's that irregular bone like the vertebrae. there's that sutural bone, right? Notice again, between the two bones of the skull. This one's a relatively large sutural bone. Here's a little smaller one over there. And then there's that largest of the sesamoid bones, our patella. All right, so 6 major classifications, structural classifications.
Question.
Yes.
You said it's not necessary that everyone might have a sutural bone, correct? Or is it just that how many you have is different?
What I would say is that I have never seen a skull that doesn't have at least one sutural bone. Most have dozens,
Okay.
but everybody gets them in different locations, and they're going to be different sizes in different locations. So there's no way to be able to say, right, this one's Bob and this one's Henry and this one's Steve, because while everybody has them in different locations.
Question.
Right? Yes.
So we're always going to have the sutures, but not everybody will have the sutural bone, correct?
Like I said, every skull I've ever seen has sutural bones in them, but in different locations. So
Gotcha.
is everybody going to have this sutural bone in this particular suture? No. No, that won't be the case, right? But that's weird. Everybody will have sutures. Sutures are what hold the bones of the skull together. But where people have the bones in between the sutures is going to be different from person to person. Okay?
Side question. You said what was the reason why we don't have more named bones? Because you said there are more, we just haven't named them. What was it?
Right. So notice, okay, everybody has this bone right here.
Mm-hmm.
This bone right here is a bone that everybody has, so we call it the occipital bone, right? And everybody has that bone, and that is one of the bones that we've given a name for because everybody has it. Everybody has a humerus. Everybody has a parietal bone, and so on and so forth. However, not everybody has this tiny little bone right here in this location, right? Maybe you have this bone here. Maybe I have a different sutural bone that's the same size up here. Maybe somebody else has one over here that's even larger. Maybe someone else has one in the exact same place, but it's much, much smaller, right? Because they are not consistent between people, it wouldn't be meaningful to give this little bone a name if only 2% of the population have it. So this is a sutural bone. It is a bone, but it's not a bone that is consistent across people. Everybody has a parietal bone. Everybody has an occipital bone. Everybody has a humerus. So we're able to give those bones names.
So the sutural bones, so we name them generally, but we don't give them a specific name.
Correct.
Location. That's the only thing that's missing,
Correct.
right?
No sutural bones have specific names. Most sesamoid bones do not have specific names. There are some exceptions, and the patella is the most obvious, right? Most people, nobody spoke up, so that means everybody has 2 patellas. That is an example of a very large sesamoid bone, but because most people have it, they've given it a name. I may have a second sesamoid bone here in this particular tendon of my wrist. Wrist sesamoid bones are common, but maybe not in the exact same location, maybe not the exact same shape, right? Maybe only 10% of the population have this one here, so we can't give it a meaningful name. So again, we just give most sesamoid bones general name, sesamoid, all sutural bones general name, sutural bones. But all the long bones, all the irregular bones, all the flat bones, all the short bones are consistent between people. So every single long bone, every single flat bone, every single irregular bone, every single short bone has a specific name. Okay? Yes.
What about the auditory bones? Where would those fall under?
The auditory bones are examples of irregular bones.
Okay.
Yeah, the auditory ossicles are irregular. All right, excellent. How are we doing? We are doing good. Okay, excellent. So we'll go ahead and take our next break at this point here because we are going to now talk more about the specific anatomy of these specific types. So here are the structural classifications, but obviously they have specific anatomy that is going to be different from each other. So in particular, we are going to talk about the specific anatomy of the long bones. And then, because they're all kind of similar, we are going to take the specific anatomy of the flat bones, the short bones, and the irregular bones and lump them together because their anatomy is kind of consistent. So we're going to look first at the long bone because this is the one that has the most anatomy to it. And then we'll look at the basic anatomy of the rest of these bones and compare those and talk about those. And then once we've identified the types of bones, then we can finally start talking about bone features. And that will lead us to today's lab. All right, questions on that? All right, yes, go ahead.
Are we going to go back to the CRC lab thing you had up for a minute or two? Are we going to look at it again or no? Because I had a question about that.
Oh, what was your question?
It was just that when we had the arrows were pointing and all that stuff, what is the distinction that one area is, like what's the distinction or the boundary between the 2 or 3 areas that are labeled? Like you showed the picture of the skull and they had named all the areas. It just didn't say which one ends where and which one begins where kind of thing.
Well, typically the sutures are what define the bones, right? That's part of the challenge of what you're going to have to do is you're going to have to, absolutely. So as we, let's see if we can slink back to that, see if it's still up. Where is that? Here. So when you, for instance, look at the skull, you can see the sutures. So for instance, this is the sutures that connect this bone together that represent and isolate the nasal bone. So this is the nasal bone. Right here we can see the sutures that separates the 2 maxillary bones. Right, so yeah, as you look at this, as you get used to looking at these things, you're going to need to be able to recognize and identify where the individual bones are on this. Now, as you're learning this, as you look in your textbook in your lab manual have some colorized photos where each bone is individually colorized, and you can start using that to study this material. But on the exam, you're going to have pictures just like this, and just like this picture, I could have arrows pointing at all of these things, and you would need to be able to identify what these individual things are. Absolutely. All right, either these bones or bone features. All right, now, remember, you have a study guide, or is every single thing that CRC has labeled on their picture necessarily something that you're going to be responsible for on my exam? No, the mental foramen, you'll notice, is not one of the features that's on there. So when you're studying these things, it is important to have your study guide present so you're not wasting time learning things that aren't going to be on your exam. Ignore the things that are not on your exam and focus on the things that are. Okay? All right, any other questions? All right, let's go ahead and take our next break. We'll again take a 15-minute break, so come back at 10:37, and at 10:37 we will pick up from there. All righty, so I'll expect a little bit more interaction as we move forward on this, but now that we have our 6 structural classifications, like I said, we have to focus on the anatomy. One of the bone types that has probably the most elaborate anatomy is the long bone. So again, one of the things we're responsible for is the gross anatomy of the long bone. And as always, using appropriate anatomical terms. As we talked about, a long bone is typically a thin, narrow bone that has a long, narrow shaft and 2 enlarged heads to it as well. But as was already pointed out earlier, we're not going to get away with terms like head and shaft when we're talking about the long bone. So instead, as was mentioned, the appropriate anatomical term for the shaft region of a long bone is a diaphysis. So that is, all long bones have a thin, narrow, elongated diaphysis. And because I don't think it's been said yet, what is the appropriate terms for the enlarged head regions of the long bone? This is the part where you respond. There you go, epiphysis. Excellent. So epiphysis, singular IS, epiphyses, ES is the plural. Excellent. We do have one issue though. We have two epiphyses. So how do we distinguish the two from each other? How are we going to tell one epiphysis from the other epiphysis? How do we distinguish them? Anyone know?
Proximal and distal.
Perfect, excellent. Remember, one of the things we talked about is all of the long bones are in the appendages. And when we talk about the appendages, we talk about things being closer to the core of the body and further from the core of the body. So the ones that are closer to the core of the body are the proximal epiphysis, and that would make the other one the distal. Oops, I spelled distal right. Excellent. Proximal epiphysis and distal epiphysis. Perfect. Now, not only are the names for these regions of the bone different, but there are some anatomical differences to them as well. Our diaphysis is filled with thick, compact bone, but it is not completely solid. As we saw in the illustration before, there is a hollow cavity at the middle of the diaphysis. And that hollow cavity, as we saw, is going to be lined by the spongy bone. So there's spongy bone in here. And as we learned in the last one, right, when we have this outer portion and inner portion, right, a candy-coated shell and a chewy nougat center, what was the term we used when we were talking about the hair for the central region? Medulla, excellent. So what we have here is the medullary cavity. This medullary cavity, as we talked about, is lined with spongy bone. And I think I'm going to decrease the size of this font to make it fit in here a little bit easier. Excellent, is lined with spongy bone, and it contains, in a mature adult, what would it contain inside of this medullary cavity?
Yellow bone marrow.
Excellent. Perfect, it would contain yellow bone marrow within it. Excellent. Our epiphyses, on the other hand, have a very thin layer of compact bone. So again, we have a thin layer of compact bone. Oops. And instead have a large amount, and again, I'm just going to do it as a hash mark type of thing, but remember, this is all irregular in its shape. It's not linear and regular like I've drawn it here, but it has a large amount of spongy bone. Which contains within the spaces. What are contained within the spaces of the spongy bone?
Red bone marrow.
Red bone marrow. Excellent. I don't know if anybody here has donated bone marrow before, but it is an excruciatingly painful process because of where the bone marrow is located. You just can't go into the easy part of the diaphysis of the bone. Typically where they go is into the head of the femur. The femur is the largest bone. The proximal head is one of the largest epiphyses. So they will go in there, or sometimes they'll go into the coxal bone, the hip bone, which as we'll see is an irregular bone, with a very large bore needle. Yes, they anesthetize the area, but still they're taking this massive large bore needle, penetrating it into the head of the bone so that they can extract out a very small amount of bone marrow. Typically, to get enough bone marrow for a transplant, they have to do this 4, 6, 10 times with this huge, massive needle into the bone to extract it. Like I said, an incredibly sore and painful process, and an incredible generous gift for someone to be able to give. So donating bone marrow is not something people take lightly, but it is an amazing gift for those that do it. All right.
Quick question.
Yes.
So if you do donate the bone marrow, I'm assuming that it does get replenished by itself, correct?
Yes. Again, you have those mesenchymal cells that will replace the bone marrow that has been extracted. Absolutely.
Okay.
Yep. All right. So we have these two very different anatomies to this region. And so there is also, not surprisingly, a neck region. I'm going to change the color here. A transition region where you transition from the anatomy of the diaphysis to the anatomy of the epiphysis, what we would commonly refer to as the neck of the bone. But again, we're not going to get away with terms like neck. So what would the term that we would use for that transition region be? There you go, excellent, metaphysis. So we have the metaphysis, and again, we would have two of them, so we would distinguish them by the distal metaphysis and the proximal metaphysis. Excellent. Perfect. Now, these are new terms that we haven't talked about before, but let's talk about some of the things that we do know that is going to be associated with it. So let's pick a different color this time. Well, let's go with green. As we know, there is on the outer surface of the bone a dense, irregular connective tissue that lines the surface. And so, of course, this dense, irregular connective tissue is the tissue type. But because it is on the outer surface of the bone, based on its location, what did we say we call it again? Periosteum, excellent. Perfect. Periosteum on the outer surface, housing immature cells. But we also mentioned that there is going to be one lining the spongy bone of our medullary cavity. Once again, this tissue type is a dense, irregular connective tissue. Again, if we peeled this off of the inside of the bone, peeled the one off the outside of the bone, peeled them both off and looked at them under the microscope, they would be identical. They're both dense, irregular connective tissues. Where they're different is their location. So the one that lines the medullary cavity, based on its location, we refer to it as the endosteum. Excellent, endosteum, perfect. We also know, what should I use for this? I'll use, what color haven't I used yet? Pink works, excellent. We also know that while the periosteum covers most of the outer surface, we do know that on the edges of the bones, where the bone is going to form joints, there is another type of connective tissue on that outer surface. And what type of connective tissue was this again that was on the outer surface? Well, close. You've got the right idea. This, oh, don't want to do that. Let's undo that. Let's go back there. Let's do that. Excellent. You are correct. Perfect. Excellent. You're both absolutely correct. The tissue type is hyaline cartilage. But as was mentioned, hyaline cartilage is located in lots of places inside of the body. This particular one happens to be on the ends of the bones to form those joints. So we can think of it as based on its location, or you can think of it based on its function. We refer to it as articular cartilage. Articular means to form a joint, right? Your joints are your articulations. So it's articular cartilage because it is the cartilage that forms the joint. So we could think of it as its name based on its location, or we can think of it as its name based on its function. It is made of the hyaline cartilage tissue, right? Hyaline cartilage connective tissue, but based on its function, based on its location, we refer to it as articular cartilage.
And that would be a point off if you mentioned it to be hyaline cartilage, correct, on the exam?
Yes, if I asked you to identify this tissue based on its location, and in this case, I would probably say in parentheses based on its function, articular cartilage. If you say hyaline cartilage, again, you're providing me with information, you're providing me with accurate information. This is indeed hyaline cartilage. The problem is you're not answering the question. And if you're not answering the question, I can't give you full credit for that. So if I ask for the tissue type, the answer is hyaline cartilage. If I ask for the tissue based on its location, it's the articular cartilage. And it's the same thing with the periosteum and the endosteum. If I used either of these two arrows and asked you to identify the tissue type, the answer for these would both be the same, dense irregular connective tissues. However, if I asked you to identify the tissues based on their location, the answers to these two would be different, right? So like I said, I am very precise in the wording of my questions because I want you to be very precise in your answering of them. And that right there is the perfect example as why. Because with these two arrows, if I ask one question, the answer is the same. What is this tissue type? This is a dense irregular connective tissue. This is a dense irregular connective tissue. Exact same answer. But if I ask you to identify them based on their location, this is an endosteum, this is a periosteum, different answers. So that's why I will be very precise. And again, on your handout, especially for the histology, you have those keywords in parentheses to emphasize that to you. Okay? Excellent. Almost done. Turns out the outer surface, and this is another reason why we want to refer to this as articular cartilage, because it turns out on the outer surface of the bone is not the only location where we find hyaline cartilage. It turns out within the metaphysis of our bone, there can be a large chunk of hyaline cartilage. And hold on, let me change the width of this to make it easier to draw. Perfect. Within the metaphysis of our bone, in an adolescent, in a growing adolescent, is a big chunk of hyaline cartilage that, again, based on its location, is hyaline cartilage. No, pardon me, not based on its location. Hyaline cartilage is the tissue type. But based on its location, we give it a different name. This particular structure, the tissue is comprised of hyaline cartilage, but based on its location, or we could think of it in terms of the structure, what do we call this structure formed by hyaline cartilage within the metaphysis of the bone? Excellent. What we commonly refer to it as the growth plate, right? Hopefully nobody here in this class is the same size as they were when they were born. That you were, then I feel sorry for your mom. We've all grown bigger, and our bones get longer by growing at what we commonly refer to as the growth plate, but absolutely, as someone pointed out, is the epiphyseal plate. Look at the irony of this. It is called the epiphyseal plate, but it is found in the metaphysis. Now, the reason for that is it holds the epiphysis to the diaphysis. So it's what anchors the epiphysis. That's why it's called the epiphyseal plate, but it should be really the metaphyseal plate because it's within the metaphysis of the bone. Now, as I mentioned, all of our adolescents are going to have hyaline cartilage within there because they're continuing to grow their bones longer. But every single person in this class signed their own lab waiver form, which means that you were able to, that you're 18 and were able to do it for yourself. As such, then, you're all mature adults. And is anybody still getting taller? Anybody here still growing bigger, longer? Dead silence. I'm going to guess that means no. Why not? What happens to that epiphyseal plate?
It reached maturity, or?
Yeah, and when it reaches maturity, it seals, as was mentioned. And what it means to actually seal is what actually occurs, and let's go ahead and use a different color for this. What happens is that cartilage, and I'm going to draw it on the left side just to show the difference. Again, it's not like it's going to be open on one end and closed at the other end. I'm just showing the differences here. Within the metaphysis, what will eventually happen is that epiphyseal plate will actually ossify. And when it ossifies, it basically becomes a thin, line of compact bone. And let's change that to black. Becomes a thin line of compact bone. And when it becomes a thin line of compact bone, what do we call it now? What is it based on its location, or what is that structure? Now that it is a thin line of compact bone? Epiphyseal line, excellent. Right. If you randomly came across a long bone that was just sitting out in the woods, and you'd want to figure out who it belonged to, one of the first things you would do is try to figure out the age of the individual. And the way you could tell the age of the individual is to see if that bone either had an epiphyseal plate or an epiphyseal line. If it has an epiphyseal plate, that typically means that the person is typically, well, depending on how thick the epiphyseal plate is, if it's really thick, then typically that person is under 18. If it's reduced in size, then they're typically between 18 and 25. And if it's closed to a line, then they're typically older than 25. So just the presence of whether there's cartilage, how much cartilage, or if it's completely sealed, gives us a general idea of the age of the individual. So we can tell something about the age of an individual just by this one characteristic of their bones. All right. Excellent. I think we've hit all the highlights. Again, with my amazing drawing skills, have done an amazing and incredible job of drawing this. But let's go back to the pretty picture from your textbook and see what that looks like. See if we got everything. Here is the picture from your textbook showing an actual long bone. Again, as we talked about, the shaft region is the diaphysis. We have the two epiphyses that we distinguish as being proximal, closest to the core of the body, distal, further from the core of the body. Notice we have that articular cartilage on the outer surfaces. And what type of cartilage did we say that was? What tissue type is that again?
Highland?
Highland cartilage, excellent, perfect. And then we have these transition regions, the metaphyses, the neck-like region, where we go from that hollow diaphysis with the medullary cavity to the completely spongy and a thick compact bone outer shell, to a very thin compact bone outer shell, and a massive amount of spongy bone. Notice the difference of the bone orientation helps in its function. This big, thick shaft gives it that resistance of the shearing forces, whereas this compact bone gives a lot of structure and support from multiple axes, right? The head can resist shearing forces, compression from multiple axes, whereas the bone, it's typically arranged along the osteons. And again, in that metaphysis, we're either going to have an epiphyseal plate, a thick piece of hyaline cartilage that allows for growth, or a thin line of compact bone that is going to be that growth plate has sealed, and that bone is no longer getting longer. We have a periosteum on the outer surface, which is that dense irregular connective tissue. And remember, we talked about it having three functions. Houses the immature cells and blood vessels, acts as an attachment point for tendons and ligaments, and protects the bone. We have, there you go, perfect. The medullary cavity, lined by spongy bone, containing yellow bone marrow, and it is lined with that endosteum. Again, it's not going to be an attachment point. It's not going to provide protection, but it can still house those immature bones. All right, I'm sorry, hold on 1 second. All right. Questions on that? All right. If we're comfortable with this, that's the anatomy of the long bone. Again, definitely testable material that you're responsible for. We also need to talk about both the periosteum and the endosteum, remember, are both dense irregular connective tissues. Right? Remember, the only difference between the two of them are where they're located. One on the outer surface, one on the inner surface. All righty. So the long bone has a very elaborate anatomy to it. The short bones, the flat bones, and the irregular bones have a much more basic anatomy as we see here. Notice here we are looking at a long bone, a flat bone, but it's going to be the same for short. It's going to be the same for irregular. So I have enough room here where I'm actually just going to draw it here as well. So here's the flat bone. I'm going to cheat and draw a short bone. A short bone, as we can see with our flat bone, has a thin layer of compact bone. This thin layer of compact bone forms the entire outside of it. And because it's on the outside, as we learned when we were talking about the hair, we refer to it as the cortex. So remember, we had a medulla and a cortex in our hair. Here in bones, we have a medulla and a cortex. They're just in different types of bones. The long bones have the medulla. The short bones, the flat bones, the irregular bones have the cortex. Now, as you can see on the inside, we are going to have, and let me make this a little thicker to make it easier, all these irregular points and processes that form the bone type spongy bone. So again, inside here, where's my arrow? Perfect. That's too big. Inside here, we have our spongy bone. And again, spongy bone is the tissue type. Spongy bone, connective tissue, right? It's the specific type of tissue. Remember, it's organizing structures. Is the trabeculae. And those trabeculae are comprised of irregular layers of matrix that are called interstitial lamellae. Excellent. So again, whether you're looking at my drawing or whether we're looking at the pretty picture from the textbook, again, all of these structures are called trabeculae. They comprise the spongy bone at the center, and they're made up of interstitial lamellae. And notice there is a large amount of space. This space inside of the spongy bone is where we find our red bone marrow. So red bone marrow is both in the epiphyses of our long bones, but is also typically found in our flat bones, in our short bones, and in our irregular bones. So remember, as I said, for that bone marrow donation, they can either go to the head of the femur or either your hip bone, your coxa. All right? Now, the good news is, for the most part, that's it for the anatomy, except for one pesky thing, right? If you've learned nothing at this point in the class, and hopefully you've learned something, but if you've learned nothing else at this end of the class, hopefully one of the important things that you've learned is that anatomists hate you. All right, there are lots of ways they show that, and here's one of those examples. All of the anatomy, notice that we've just talked about, the cortex, the spongy bone, the trabeculae, the red bone marrow, is true for flat bones, short bones, and irregular bones. However, in flat bones only, and only in flat bones. And don't ask me why. I don't know why. This is just because anatomists hate you. That's why. In flat bones and flat bones only, they give the trabeculae a special name. So a trabeculae, or trabecula, singular, or let's say it this way, the trabeculae in a flat bone, right, based on its location, because it is in a flat bone and only because it is in a flat bone, as you can see, is called diploe. We had the little umlaut over the top. Again, I don't know why. I don't know why in just a flat bone, but it's one of those pesky things that they have done, and I don't have a good reason for why. But we need to know it. So in a flat bone and in a flat bone only, the trabeculae of the spongy bone, based on their location, are referred to as diploe. So just put that in your pocket, right? Maybe you'll get to know that for a trivia question sometime on some trivia show, but I don't have any other reason why you need to know that other than that's what it's called and we need to know that.
Question.
Yes.
Do we need to know this for the exam? And if we do we need to put the little dots above the E?
No, you don't have to put the little dots above the E. If you were writing it by hand, then I would say, yes, put the little dots above the E. But since you're just typing it, don't worry about changing your keyboard so that you can do the little umlaut on top. No, just write it out DIPLOE and that'll be fine.
Okay.
Okay. And so, yes, absolutely. If I showed you an image like this or an actual cross section through a flat bone and I had an arrow pointing to this structure, right? Again, and let's make that bigger and easier to see. So undo that, do that, do that, right? And I did this. If you think about it, there are lots of questions I could ask. I could ask you to identify the tissue type, be specific, and you would say spongy bone. I could ask you to identify the organizing structure, and you would say trabecula. I could ask you to identify the type of layer of matrix that forms this structure, and you would say interstitial lamellae. And then I could also ask you, identify this structure based on its location, and because this is a flat bone, you would say diploe. So again, the arrow never moved, but I was very precise about how I worded the question, so you were equally precise in your answering it. All right. Questions on that? All right, excellent. With that, we are done with the general gross anatomy. So again, we have all the pretty words that go ahead with that. And again, this typically contains the red bone marrow. We did all of that. That's all good. That's all there. What that brings us to is the bones and bone features. As I mentioned, you have 206 named bones. And you are responsible for knowing all 206 named bones, right? You must know all 206 named bones. Also, for every single paired bones, what do I mean by paired bone? What do I mean when I say a bone is paired?
Is there where two bones connect to each other?
No, close though. Paired does have to do with two. What do we mean when I say paired bones?
Oh, is it like proximal and?
No, not necessarily. If I told you that the femur was a paired bone, why is the femur a paired bone?
Because there's two bones that make up the femur.
Say again?
Is it because there's two bones that make up the femur?
Well, it's not that there are two bones that make up the femur, it's that you have two of them. A paired bone is a bone you have two of. You have two femurs, one on the left and one on the right. And if you think about what we talked about in the last class, if you've broken your femur and they're wheeling you into the emergency room, Do you want them to flip a coin to decide which leg they're going to put the cast on? These are the easy questions. No.
No.
So when you have paired bones and we're dealing with paired bones, you must be able to identify left versus right. So you must be able to look at a femur like the one right here on the screen and realize, that is a right femur. And you must be able to identify that, right? Notice you only have single vertebrae on the midline, single mandible, so you don't have to worry about identifying a right and a left mandible, but you do have two femurs, so you need to be able to distinguish left and right, all right? So you must know all 206 named bones, and any bone that is paired, you must be able to distinguish a right one from the left one. However, the other thing you are going to be responsible for are bone features. Now, every single bone has dozens of bone features, because as we've learned, anatomists love to name everything. Are you responsible for every single bone feature that is in your photographic atlas for every single one of those 206 named bones?
Hope not.
Hope not. Well, if only there was a study guide that told you precisely what you were responsible for on this exam, you might be able to figure that out. So, anybody bother to print out the study guide like I asked? Let me ask you a question. How many bone features are you responsible for on the frontal bone? I'll wait. If you haven't printed it out, then go to your modules, look at your study guide, and tell me how many bone features of the frontal bone you are responsible for. 1. Excellent. How many bone features of the ethmoid bone are you responsible for? 7. How many bone features of the vomer are you responsible for?
1.
Do you see a bone feature for it on there? What page are the bone features of your skull bones on? What page number? That's page numbers on it, right?
2.
Page 2. Do you see the vomer on that list of bones on page 2? Do you see the vomer back on page 1?
Yeah.
Yeah. Vomer is definitely one of your bones. It is definitely a bone you're responsible for. But guess how many bone features you have to know of the vomer?
1.
None. Does that mean the vomer doesn't have bone features?
No.
No, of course it has bone features. But how many of them do you need to know for the exam?
0.
0. So all you need to do is be able to recognize the vomer. How many bone features of the nasal bone or the lacrimal bone do you need to know?
2.
Well, those are bones. Be careful. Those are 2 bones.
Right. So none.
But are there any bone features you're responsible for?
Nope.
No. And see, and that's the key. You are responsible for all named bones, being able to tell left and right when appropriate. But the key to a bone feature, and again, this is going to be silly in my definition, but a bone feature is a characteristic or a feature of a single bone. All right? So let's again look at the example up here of the picture of our femur. Our femur has this, and where's my highlighter? Our femur has this nice big round bone feature that is a feature of this bone. Anyone know what that big round bone feature happens to be called? If you want, you can cheat and look ahead on your study guide for the femur and guess.
The ball socket.
Close. It forms a ball and socket joint, but the name of the bone feature is the head. It's the head of the femur. Okay? Not all of them are going to be that easy, but that is a characteristic. It is a characteristic of a single bone. if I have a bone in my hand, what do I do with my pencils that I had here earlier? Excellent. All right. This clicky thing right here is a characteristic, a feature of this pen. When if I hold the bone in my hand, if I hold a femur in my hand, it has that big round head. So that is a characteristic of that bone. And what you have on your list is a precise list of the specific bone features you are responsible for each bone. And some bones, the answer to that is none. You don't have to know any bone features of them, not because they don't have them, but because they're just characteristics. The frontal bone, you only have one. Doesn't mean it just has one bone feature. There's only one you're responsible for. I make a point of emphasizing what a bone feature is because the other thing you'll find in our skeleton are what we call skeletal features. Actually, I should capitalize this too. A skeletal feature is a characteristic that is formed by two or more bones. All right. One of the things that I do first thing in the morning when I wake up, even before I brush my teeth, is I make sure I put a little rouge on my cheekbones. Right? Okay, not really, but right, I do have these cheekbones that we think about, right? Nice, beautiful cheekbones. But guess what? There is no bone called cheek. In fact, what that cheekbone is a structure that is referred to as the zygomatic arch. The zygomatic arch is, as the name would indicate, an arch. And that arch is actually made by two bones. Notice, if I just have one bone, I don't have an arch. I do have a bone with a thing that sticks out, and this thing that sticks out is a bone feature of this bone. And then I have a second bone that has a bone feature. But when I put those two bone features together, I suddenly have a skeletal feature. That arch is made by two or more bones. And so that we would call a skeletal feature. Now, as it turns out, your skull has a lot of these features. So again, because anatomists love vocabulary, we will talk about, and you have a list of skull features you are responsible for. But really a skull feature, all a skull feature is a skeletal feature of the skull. Or we can be more specific in our definition. We could say that a skull feature, oops, oh no. This one here, do that there, perfect. A skull feature is a characteristic of the skull that is formed by two or more bones. So really, it's just a skeletal feature of the skull, but there's so many of them, we use the term skull feature. There are dozens of them. How many skull features are you responsible for?
24.
Is there 24 on that list? That sounds about right. Yeah, I didn't actually count, and I don't actually have the list in front of me, but that sounds like a good number. Something like that. You have a list of them there on page 2 as well. And those we'll go through together. All right? So what we are going to be talking about are now characteristics of the bone and characteristics of the skeleton. These are some confusing terms, these bone features and skeletal feature terms, right? Things like trochanters and tubercles and tuberosities and condyles and processes and all these things. And as random as it seems, there is some meaning and method to these. Typically, bone features are things like elevations, things that stick off of a bone, or attachment points. An attachment point can be where a muscle or a tendon attaches to it. Articulation points, where the bones form joints. Depressions is an indentation, and an opening or a hole, right? An opening we could also think of as a hole, right? A depression is an indentation, whereas an opening would be a hole through that. All of these things have names to them, specific names that mean something. So like, for instance, a tuberosity is a large rough attachment point. That vocabulary matters. Your book has a nice table that talks about these terms, and you should know the definitions of these terms. You should know that a condyle is typically a smooth, oval, articulating surface. Because then when you're looking at these characteristics on the bone, you'll be able to easily tell the difference between a condyle and a trochanter and a tuberosity. So learn this vocabulary so that you can make more sense of these. So again, if you want to just brute force memorize all the terms, that's fine too. But understanding what these terms actually mean will help you when you're confused and trying to remember what it is. You can see, this is a large, rough attachment point. This, for a muscle, this must be a tuberosity. All right?
Are we going to be going over the bones, or just you need to memorize them?
We are going to go over the bones, and we're going to go over the bone features on your list. But the key word there is we. What we are going to do right now, excellent, we're doing great on time. What we are going to do now is we're going to take our last break. When we come back from our break, I am going to be breaking you guys up into individual groups of 2 or 3 people. You are going to be put into a breakout group here in Zoom. And I will also be adding you to your own individual group discussion boards on Canvas. What you are going to be is using the images that I have provided for you. You are going to be going through these, and you're going to be given a bone and bone features, or bones and bone features, that you're going to be responsible for as a group presenting. Now, again, you're not going to be graded on your presentation skills. You can don't have to have this memorized. You can have it all written down. Hell, if you don't want to have your camera on, you don't have to have your camera on. But what I'm going to provide you for is some images. On those images, or you can use your own images. You can draw your own images if you want, however you want to do it. But what you're going to do, the same way that this picture here has bone features labeled on it, you are going to draw lines and write out, label these pictures, and then one at a time where you're going to present your picture. We're going to talk about the picture. We'll talk about some of the functions of some of these features and identify them, and we'll go through them one at a time. So what I'm going to do is break you out in the group so you can start discussing how you're going to do this, how you want to do this, what's easiest for you. Like I said, so you guys can compare contact information. I'm going to set up a discussion board for you so you guys can communicate asynchronously. But between now and Thursday, you are going to be responsible for labeling a certain number of bones, and we're going to start on the axial skeleton first. And it's going to be real simple. We're going to go straight through in order on that handout. So whoever has the frontal bone goes first. What's the second bone? Come on, what's the second bone on your list?
Ethmoid.
Ethmoid. All right, what's the third bone on your list?
Sphenoid.
Sphenoid, excellent. So there you go. We're going to go right through order. So whoever has the frontal bone is going to go first. They're going to present us our one bone feature, right? The sinus, we'll talk about it briefly. We'll laugh, we'll cry. Then we'll move on to the next group, and they'll do it. And then we'll move on to the sphenoid, and we'll do it. And we're going to go through the list. Our goal for Thursday is to do the entire skull. And once we're done with the skull, I will help you put these pieces together to make these skull features that you're responsible for. Then on Friday, we will do the rest of the axial skeleton. So those will be the groups that have the vertebrae, the groups that have the sternum and the ribs. And also on Thursday, you will be given your appendicular assignment because you'll be presenting twice, once for the axial skeleton, once for the appendicular skeleton. This is a graded assignment where I expect you to work together as a group. All right, to present this. If only one person wants to talk, that's fine. If you want to split up the bone features so you each present 3 of them, that's fine. However you want to do that, that is going to be fine for doing it that way. You guys can decide how best you want to do it, but you will be getting a group grade for correctly completing this activity. Right? And again, if you get something labeled wrong, that's not the point. Again, this is the point of this is to be a learning experience. So as long as you participate in this activity, you'll get the full points. You're not getting graded on your presentation skills or anything along those lines, or your laboring skills or your drawing skills or anything like that. This is a participation grade for participating in this activity, because the point of this is to make it a more interactive learning process. So I'm not just going through, this is the greater trochanter, this is the lesser trochanter, this is the intertrochanteric line, and so on and so forth. That's incredibly boring, incredibly monotonous. So this makes the process a little bit funner. Maybe not funner, but more interactive anyway. All right, so it's gonna take me a little bit of time to get this set up and form our groups. Let's go ahead and take our last break. We'll call it a 13-minute break, because then I'm gonna break you into groups. Give me to 11:35, and then that gives you guys an hour to work together, or a little under an hour, because then we'll come back together at the end to see if there's any questions. I'll pop between the groups to see if there's any questions as well. But let's come back, let's try to come back at 11:35. I may close this window, so I'm just gonna write this down here so that I remember. 11:35, and at 11:35 we will pick up, we'll look at the materials, talk about the materials, form our breakout groups, and go from there. All right, I will see you at 11:35. So let's do Apparently I didn't pause the recording, didn't I?